Good morning, everyone. Morning, everyone. Morning, everyone. Morning, everyone. Jack Leckerman, we firmly believe in the magic of cooperation and partnership. That is why I want to personally thank to all the speakers and also the sponsors for their support during the preparation and organization. To Citiva, Merck, Sartorius, Paul, Thermo Fisher, Terumo, Jack Leckerman, and Unisima. We firmly believe in the magic of cooperation and partnership. To our partners, that is why I want to personally thank to all the speakers and also the sponsors for their support during the preparation and organization. To Citiva, Merck, Genéricos, Sartorius, Alanac, Avifina, Thermo Fisher, Unifarm, Jack Leckerman, SCI Publishers, World Pharma. We firmly believe in the magic of cooperation and partnership. To our partners, that is why I want to personally thank to all the speakers and also the sponsors for their support during the preparation and organization. Welcome to the Biosimilars LATAM. I hope you enjoyed the program, and I don't want to forget about our virtual participants. I hope you get the most out of the forum from the comfort of your office or home. Now and finally, I want to invite to the stage to our chairman, Marcelo Holanda, the head of LATAM in Biocon Biologics, Brazil. Marcelo, welcome, and the stage is yours.
So, good morning and welcome to São Paulo. Bom dia, bem-vindos a São Paulo, minha cidade, onde eu nasci. También para los que hablan español, bienvenidos a São Paulo. Un placer tener todos acá juntos.
Recording in progress.
So, I'd just like to thank you, everybody, to join us. We are talking this week about these 2 days about biosimilars. So Biosimilars Forum, for me, is a very special topic. It's to think about 25 years ago. We don't know where's this future for looks like for biosimilars. There is a lot of uncertainties about production, about quality, about regulation. And now, 2022, we know how important those products, because at the end of the day, what is the most important? Patients need those products. We need to increase the access in Latin America. We need to increase the access for patients. And access is not about price, it's about the quality. With a couple of colleagues that I know here, we have this kind of the discussion before, and we think about, if I need to use the product, and I hope we don't need to use a pharmaceutical product in the next coming years, but if I need to use the product, should I use the biosimilar? If my mother my father, or someone from my family need to use the product. Should I recommend the biosimilar? So yes, of course, because biosimilar is a reality in Brazil, is a reality in the world, is a reality in Latin America. We need to have the products that go from the development, best quality service, best quality equipment, best quality development, and also best clinical trials that prove the safety for the product. It's a different market. Not about the generic. It's a very different market. But we need to have the regulation. We need to have this. So these 2 days is about that. We have the best speakers that we can put here in front of you to talk about this. We go on the edge of regulation. We go and we understand the best of a production. We go and talk about the tender market. And we wanted to conclude this in 2 days that we at least think about what is the future of biosimilar in the next 10 years. So that's the idea. So more and more products, more and more biosimilars will arrive in the market. And you need to make sure that we are able to use that in a safe way. We are comfortable, can share this. I'm very happy to be here and share those moments with you. The opportunity that I have here is not only about to invite you to a coffee, invite you to have a lunch, but is provoke you to ask questions. That's great opportunity that we have. We have a group of people here that can raise good questions during this discussion that we have. So I wanted to invite you to really pay attention. Shut down your mobile phone for a while. Your boss is not calling you in the next 2 or 3 hours. And let's make sure we can take advantage to be together. To be together face to face is a new word for us now. We have a lot of people that are connected through Microsoft Teams, to Zoom, but let's take advantage to be together again at this moment. So I have a couple of comments here, basically to say something about how this flow of days is going to be. The first one is quite a disclaimer that we need to have. The disclaimer is about the speakers that not necessarily reflect the views and opinion about the companies or institutions that they represent. And also there is no economic exchange for the participation of anyone that is speakers from here. No one are paying for that. That's a very important disclaimer for us. The next one. I just comment about this networking. We have a great opportunity here to take some kind of networking and talk about this. I just recommend you when you finish the presentation, we have the timeline for the presentations. When you finish the presentation, you have the microphone. You can raise your hand and you can circulate the microphone and you can raise your questions. We have always 5 minutes, 10 minutes to have these questions. Feel free to ask questions also in your local language. In Portuguese, we have the translators here. So come back. And also, if you are connected with us through Zoom, raise your hand on Zoom, and then we can connect it with you. We can pick up your question by chat, and then we can read the question here. So you have the connections here to connect it with the translator. Feel free to pick up this, use the translation, because sometimes it's really important you have the translation and really understand and then get the focus on the discussion. Don't be shy to use that. So all the presentations is gonna be sent by next Friday to you. You don't need to take photos or write down. So enjoy the moment, take a look on the presentations, and we share with you all this material at the end of next Friday with the team. You share this by using the email you are here. So that's my let's say my first comments about that, and I wanted to invite him for our first session here, Dr. Merusa Freitas, sorry, that is from Anvisa. She has a lot of experience on Anvisa. It's gonna be great to have someone from Anvisa here to talk about the latest development on the regulation. So, por favor. I don't know why I said Freitas, Freitas. Thank you so much for being here.
Good morning. Bom dia. Welcome.
Good morning.
Bem-vindos ao Brasil. A minha palestra, eu preciso falar em português.
So I will need to speak Portuguese.
Eu vou abordar as questões regulatórias
as it's important to speak Portuguese in my country. So I'll speak about regulatory matters, especially how Anvisa has been dealing with biosimilars, which are such an important product to access new therapies and also broaden access. Once a biosimilar reaches the Brazilian market, it usually comes at a much lower cost. So it's a relevant therapeutic option, especially for therapies that are so expensive. And they are regularized in Brazil through biocomparability. So thank you for inviting Anvisa to this important forum. And I wish I could be here for the entire event, but unfortunately I will need to leave to help with some health matters, as in the next few hours we will assess a new drug that will reduce the damage that COVID has caused to Brazil. So moving on to the topic at hand. I skipped through a few slides here. So
okay, thanks.
I will bring some of the latest news on Anvisa's regulatory processes for biosimilars in Brazil. Recently, the World Health Organization published a study with data and a survey from the pharma industry since 2019 and 2020. I believe that the challenges that they laid out are still very pertinent, still in 2022. But I also have to mention that we had significant advances since 2019, since that first survey from the WHO. And that is true as we see that many products have been approved through the biocomparability pathway. But the challenges that we have now are different from 2010. In 2010, we still had some problems with terminology, biogenerics. What trials will be accepted for biocomparability? There were some medications that were used in the biosimilars pathway that were not biosimilars per se. Furthermore, we have seen major advances, but I have to confess that we expected to make a bigger stride. We wish we had more products. And the survey from the World Health Organization includes a regulatory perspective to see what challenges need to be overcome so that we can expand access and the approval of biosimilars in Brazil and around the world. Recently, a survey was conducted in the BRICS countries, and its conclusions were similar to what we see in this survey from the WHO. Some challenges are a bit different when it comes to the BRIC countries, but the first challenge about reference products is still very pertinent. So it's a common challenge that we see in other countries as well. First, there is a challenge in finding in enough amount in different batches these reference products for comparability trials, and this is a requirement. We're going to face an even bigger challenge in Brazil than in some countries. Especially when you have a public healthcare system. Oftentimes, these reference products are acquired for public health programs, and the companies that want to develop biosimilars have trouble accessing them. Another challenge is that often some countries require a reference product to be acquired from within the country. So the reference product should come from the country itself, and authorities need to have discussions on accepting references from international suppliers. It needs to be approved, if you are coming from countries that have similar regulations in order to avoid unnecessary trials, which would increase the cost to develop these products. We know that these products require investments, and their cost is higher than developing generic because they require clinical trials, differently from generic biocomparability trials. Although there are some human studies in generics here, usually you need larger sample sizes, which makes it more difficult and more expensive. Excuse me. Okay, I'll carry on. So again, related to reference products, and this especially affects BRICS countries. Russia and India have some challenges in selecting a reference biological product, and there's even a need for them to be selected as reference by their authorities. In Brazil, we are a bit more advanced there. We accept reference products from other countries as long as the product has been authorized or assessed by authorities that are aligned with our own requirements for control and efficacy. This survey also refers to a lack of human resources. So both from a regulatory perspective, human resources working for the government, but also in the production sector for research and development, there is a lack of human resources that are trained in biotechnology. And on the regulatory side, you also need a capable team that can assess data and avoid any double work, especially to prevent against requesting unnecessary studies, toxicology studies, preclinical studies, which oftentimes can be avoided by using other information from other authorities. So the human resources challenge appears in the regulated side, but also in the production side. We also have problems with the quality of some biosimilars. This also means there are regulatory requirements, so there's a lack of clarity, and what requirements are necessary for proving the quality and similarity of a biosimilar and a new biological product, and also challenges with the production process. I mentioned that the biotechnology process requires specialized information in its production and control to avoid having a product with a higher toxicity, with impurities that can lead even to a safety risk for the patient. And I'd like to remind you that, well, related to quality, we're talking about medications that are produced with recombinant DNA. They are pooled, and we use cells such as mammal cells or bacterial cells for a protein to be produced. So that requires a lot of control to avoid any variability of the product so that it can keep a similar profile. There's another discussion, which I don't believe is so significant in Brazil, but about the interchangeability of biological products and also their nomenclature. The World Health Organization is often criticized about naming and labeling. There should be more harmonized naming standards. The FDA in the US has its own nomenclature. They have a specific guide for interchangeability, but many say that you cannot have a comparison in interchangeability according to the FDA's requirements, because that would rule out many products. In countries that have regulated interchangeability are Iran and Japan, and other countries use the strategy of manufacturing, doing their own work and showing it to prescribers by proving the quality of their products and showing that they are an important therapeutic alternative. We believe that that's the right path. Interchangeability should not prevent a biosimilar from being approved, but in naming, there should be some advances so that the entire world can be on the same page on how biosimilars are named. So I think these are the 4 most significant challenges according to the World Health Organization. And I am sure that they analyzed other countries as well, because they also refer to the realities in Brazil. Brazil has advanced. In the past, we had problems in developing preclinical studies, but companies have advanced there, and companies that are getting approved in Brazil have been able to do it. And the biggest challenge we see are in clinical trials and not preclinical trials.
Ok, thank you. Então,
So, how to solve these issues? What's the best strategy to solve them? Anvisa strongly believes that one of the ways to solve these problems, in addition to direct interactions with companies in Brazil, is working with regulatory cooperations with other countries and other authorities, believing the need of duplicating efforts, duplicating the necessary studies, working with other authorities, so you also have good knowledge of the reference ingredient, and work with joint revision. I can mention the project ORBS. That's not really for biosimilars, but we have another project, ACTS, that consider generic medicines and biosimilars. But in order to do so, companies have to be more harmonized with continuous submission of the files to Brazil and other countries and our authorities. So we can do this revision together with other authorities, increasing our dialogue. And this is a facilitated pathway that accelerates the process. Another important factor is that for the Brazilian companies that develop biosimilars, take the products to other markets, counting with Anvisa evaluation that is that has a meaning and a value in the revision by other country authorities. And the exchange of information with other authorities happen. We have interactions through MOUs and frequent reports, using all these channels so we can improve communication and knowledge on the medications we have in the market. And finally, the other way to solve is how will we deal with biosimilar medications that were authorized out of the rules that are previously established for biosimilars. These medications also have their place and their therapeutic importance, and they have to be considered by the sanitary authorities in a different way. And I would say that this is the biggest challenge, because the world is discussing evaluating real-life data, using pharmacovigilance data, so we can authorize a product. So for biologicals that were authorized through comparison pathway, but do not fulfill the current requirements, should we do new studies? Should we start from preclinical assays? I believe that this is a question for all the authorities, and COVID taught us a lot in this process, so much so that if we didn't consider real-life data, we would still be with just one vaccine and one single indication for one single target group. Surely real-life data make authorities always seeking standards, because data have to be standardized and trackable. And monkeypox is now putting us on our knees, because we have to authorize monkeypox or pox vaccine to monkeypox, based on animal clinical assays, and discuss with data of preclinical assays, discussing phase 4. And we are still living a difficult situation, and we have to seek the best way out and bring to the country the best healthcare attention. As a director at Anvisa, I passed through all of that, and I know that is a moment for reflecting and reinventing knowledge. We will have to consider more and more real-life data, models in phase 4 study, follow-up products, and surely biosimilar are the case, biosimilars that are authorized in other conditions, and in the old regulatory framework has to be reconsidered. Next, please. Thank you. So this is Brazilian landscape right now. I would say that we are not that bad considering other countries in the world. When I see that the United States has 35 products registered through FDA, and Europe 36 by EMA. And so our reality is not that bad. Not a long ago, if I'm not wrong, 2017-18, European Union have 58 products of 6 molecules. And up to 2020, we see a major expansion of biosimilars in Europe, especially due to the to the end of patents from 20th century. But there is a progression of patent for 2030. So this will change a lot what we will receive as biosimilars, because the end of patents, although the market is not as hot as we would expect, we all believe that it is a market in expansion of about, we projected for 15.8 billion, and now it is about 46 billion projection. So I believe that Anvisa will be challenged by the market. We have 46 approved biosimilars from different APIs. We have products, many of them monoclonal antibodies, but we have also a high level of rejection. 13 processes were rejected. Files, processes that were filed at Anvisa for bioavailability comparison, mainly related to preclinical and clinical assays. We had 6 processes that were cancelled and 7 that are currently under review, and 11 waiting for review. we have to mention that COVID-19 was a major impact for Anvisa because we had to dedicate ourselves almost exclusively to COVID-19 because it was a public health emergency and we have to contribute to save lives. So I have to say that this was my main challenge, working with COVID-19 products. We have products in a queue. Everybody knows that we are a small agency. We have to reinvent ourselves on a daily basis. And we have a technical team that is reduced when compared to the main agencies, surveillance agencies in the world. But this is not a reason to lag behind. We have to reinvent ourselves. An authority that is willing to talk to any party in the world. We have to be transparent. We have to promote dialogue and have well-established processes with well-established rules and transparency. Not only do it has a value, institutional value, but it is a value for the Brazilian society as a whole. This protects our health and add value to the institution, to society, and to have a model market. Companies also claim for transparency in Anvisa. We still have a long way to go to improve that, to improve transparency and communication. But we published in our portal what are the drugs authorized by Anvisa, a summary of our evaluation, so approval or rejection. What is the technical rationale to approve or reject a certain drug? We also have a report about interchangeability of drugs. All of those used in Brazil are available at Anvisa website. And we have also a page with Brazilian legal documents. They are all available at our webpage. But to conduct all these discussions, there are regulatory agencies that are highly valued by Anvisa, which decreases costs because once used in other countries with the same rules, they have an expedite pathway to get in Brazil. It facilitates the access. And I talk every day, not only with Anvisa, but also with the external market. We have to guarantee quality and safety. It is not regulatory issues that will be a hurdle. The hurdle cannot be sanitary, just out of a regulation. We want to have convergency with the WHO. We have specialists of Anvisa that work vis-a-vis WHO. We also do it for APIs and new technologies whenever WHO calls us, and in joint inspections. I would like to highlight the importance of ICMRA. Anvisa has an active voice in this coalition that involves 32 countries, exchanging our experience. Since 2020, ICMRA is leading the countries, and Anvisa has a place in this table to discuss regulatory issues. And I believe that this is a strategy that will strengthen Brazil, having a leading role in this discussion. We are also part of the ICH Forum, and it is very important to discuss our rules with other countries. I believe that Brazil is one of the few countries in Latin America that is part in the coordination group in ICH. We are stakeholders we take part in PICS, and I would like to highlight the importance of Brazilian pharmaceutical companies. I had the honor of conducting the integration of Brazil as a member of PICS, and Brazilian companies adequate and strengthen Anvisa to be a member of PIC, which is very important to guarantee the quality of the products in the market. IPRP is also a very important forum, which is an international cooperation forum for pharmaceutical programs. Anvisa has also a leading role in this forum, and all the harmonization we seek respecting specific rules of Brazil. And what is it specific for Brazil? Epidemiology, issues related to IPAs that are more necessary to Brazil. But it won't be a specificity that will prevent the product to come to Brazil. So far, we had very clear rules about reliance. I believe that the best result is a rationalization of inspection with exchange of reports among authorities, but out of inspection, to conduct inspection, and I conducted the inspection process in 2019 and part of 2020, I believe that the process is more consolidated right now. And this improved the time to approve or reject. I would say that since the beginning, I said when I was interviewed by the National Senate, one of my targets as a director in the agency was establish the requirements for regulatory reliance and improve our regulations. Oftentimes, members of the agency and the external sector fear this strategy. They believe that will open too much our market. But I would say that it's just on the other hand, we will make our market more efficient and bring this efficiency also to Anvisa. We just published a resolution in August 10th, 2022, establishing general criteria to work with optimized processes and to establish which authority we have reliability criteria and alignment. This regulation established for all the products we work with, ranging from pesticides to cosmetic products. And every technical department in Anvisa has to prepare a standard, which is our regulating model, to establish the criteria on how this authority can recognize and inspect. I would say that today we just published the August 18th public consultation 1108. This public consultation specifies the criteria to optimize the evaluation processes for biologicals, APIs, for biologicals, for medicines, for cosmetics, and other products. These public consultations will be opened for 45 days for comments. The public consultation established as a strategy, well, the major strategy on this public consultation is that the documents prepared by an authority may be shared in Brazil to benefit the improvement of processes. And with that, we want to strengthen our regulatory capacities with our limited resources, improve our efficiency, because resources are really scarce at Anvisa, avoid duplication of efforts and reworking, expedite access to medicines, to drugs, including other medications that wouldn't be benefited by Anvisa. They will have a fast approval or reject. And with that, we improve the convergence practices among regulatory agencies. This consultation and this standard establishes the equivalency. We can evaluate documents issued by these agencies, public or not, and we establish as a measure 4 modalities. A, for a product approved by for 1 year, for less than 1 year in one country. B, at any time. C, considering the risk of the project. Here we consider low risk. We approve products in phase 2 and conducting phase 3 clinical assays. And wouldn't be modality C, products that require more follow-up. And we also want, this aims to reducing Anvisa times. Everybody knows that in Brazil, the product registration should occur below 150 days, and pre-registration 90. These are still this is still a challenge for Anvisa. But in this project, we establish possibilities of differentiated evaluation using information that we exchange with other authorities to reduce this term to 100. Is currently 180 days. We want to reduce to 150 for registration and 90 for post registration. So what do we consider as an equivalent authority? What are the requirements to establish that is equivalent? So evaluation of the authority. How aligned is the authority with Brazilian standards in terms of quality, efficacy, and safety? How this authority carries out the best review practices in their evaluations, and also their guidelines and technical documents. This measure also considers if the authority uses the guidelines published by the WHO and this International Pharmaceutical Corporation System guidelines. How do they interact? How do they exchange documents, be it English, Spanish, Portuguese? And there's no restriction of access of these documents to Brazil. And it means that just by presenting this document, product will be registered in Brazil? No, all the technical documentation of the company, be it quality, efficacy, and safety, preclinical, clinical, that have to be presented in addition to the documents presented by the other authority or provided by the other authority. The documents of these authorities have to prove that a thorough technical scientific evaluation was carried out and concluded efficacy and safety, and won't be restricted by Anvisa. And here I would like to mention United States, due to their policies and legislations, that oftentimes the documents issued by FDA are classified even for the company that asked for the approval. And this information cannot be considered secret or classified by Anvisa, including in our direct interactions with the authority. I believe that this is the authority that has the highest level of secret information or classified information.
Decisions submitted through the analysis process maintain Anvisa's autonomy. It doesn't mean that the product was, if it was approved by a different authority, that it was immediately authorized in Brazil. It needs to be authorized here as well, considering the qualities and characteristics of our country. And I'd like to make an announcement here in the biosimilars process. I think that this meme here says a lot. we have looked at that survey from the World Health Organization, and we look at the number of refusals in Brazil, and it's quite high. Anvisa works for a process. The goal is always to approve a drug and not to refuse it. To be refused is an outlier. So Anvisa will release in the next days a call notice to see what challenges companies still have to approve a biosimilar in Brazil. By October, we expect to publish this public notice for companies to join us in this conversation so that we can assess if we need to adjust any of the regulatory measures or any of the assessment procedures, because the Anvisa rules are already in line with other countries. The registration regulations are in line with ICH and the World Health Organization, but we also need to look at why this market has had so many refusals, as we said. And we also have to understand how, with patents that will expire in 2023, what effort Anvisa needs to make to increase the number of approvals in Brazil. So we would like to look at the future and think about where we're going. And I apologize for including that meme. And moving towards my conclusion, we need to build a better interaction with the industry. We also want to expand our international cooperation. So what other countries also have an interest in cooperating with us? We have already established that we are going to assess processes according to the FDA, the EMA, Switzerland, Canada, and the World Health Organization. So it is important to have closer relationships to authorities. And we do have a really good relationship with the European authorities and with Canada. But once biosimilars come to Brazil, they are brought here from several parts of the world: US, Europe, Spain, Belgium, and Italy, India as well. We see that Asia and Pacific is growing a lot. So we see that there are many countries that are manufacturing biosimilars, including Brazil. So for sustainability, we need to talk about pharmacosurveillance, because they're very similar to what we use in new products. So we need to see how we can organize real-world data and big data so that we can work with real-world evidence. In fact, we've asked a group from the ICH to discuss real-world data so that we could have a more harmonized discussion among all countries. And the European unit has established a guide, but it's still in a public hearing. And the FDA has as well. So without further ado, thank you. I'd just like to say that it's an honor for Anvisa to be included in this event. Thank you.
Quando ela faz consulta pública, indústria e as organizações não governamentais, a sociedade civil. Esse chamamento é super interessante e com certeza vai ter uma participação grande com todas as consultas públicas que a indústria faz, que a Anvisa faz, a indústria e outros atores têm oportunidade de manifestar. Eu vou abrir para pergunta agora, a gente tem uns 5 minutos para pergunta. Quem quiser fazer uma pergunta, por favor, levanta a mão. Queria saber se tem alguma. Por favor. Fala. Bom dia a todos. Luciano, da Projeto América. Eu quero agradecer ao Jack Leckerman pela oportunidade e pela realização do evento aqui em São Paulo. É um evento bastante importante para a nossa área de vestibulares. É muito importante que esse mercado consiga se desenvolver sustentável. E aqui vem a pergunta para a doutora Melise, que é saber quando que a gente vai ter uma previsão para fazer a revisão da LDC 55, que nós já temos 12 anos que ela foi publicada, e de 12 anos para cá a gente já teve uma série de alterações, a Anvisa entrou no CH, nós temos uma série de harmonizações internacionais, convergência regulatória, e é muito importante até no mesmo caminho esse edital de chamamento para fazer um trabalho na área de vestibulares, que a gente possa avaliar uma maneira também atualizar os nossos registros.
So that we can update our biosimilar registrations with a resolution or a more updated regulation. So I'd like to know when we will be able to review this norm. Thank you. Thank you for your question. I think that this public notice will also give us a good basis for revisions. As you know, Anvisa accepts basically all ICH guides, even for the approval of biologicals. It accepts the CTD model. We're still moving forward with that because, I wish it was faster, but there are also challenges for companies to submit on their ISTD, but biological dossiers are accepted in the CTD format. Besides that, all guides and ICHs for clinical and preclinical guides are accepted by the biologicals area. So, according to the 2010 regulation, many of these normatives have been accepted in 2021. The ICH updates through the same resolution that incorporates ICH guides. Obviously, there are still advances to be made in the 2010 RDC. An additional point on regulatory assessment dates. I'd also just like to give you some context on that. Assessing biosimilars is more complex. All authorities spend more time making that assessment. It's a sort of assessment that requires a lot of effort and a different sort of technical understanding. So a biosimilar dossier needs to be well made. The strategy of regulatory trust is a mechanism to optimize the assessment process, considering assessments that have already been made by similar authorities. However, there will be some biosimilars that have not been approved by other authorities, or some that are developed specifically for Brazil and from Brazil to be taken to the world. And we do have that capacity. There are companies in Brazil that can open up biosimilars to other markets. Latin America is a great market. And the fact that we have a regulatory authority that is respected and strong for the market is very good in broadening our market. So the strategies that we work with today is to have a similar model to Canada, where we also want to have more confidence in our own process. So for a company that is well established with a good history, we want to have a streamlined process considering how the company is trusted and the specific requirements and history that that company has, and how much that company has communicated with Anvisa throughout the last 20 years. You can't close your eyes today and consider that you don't know the pharmaceutical market in Brazil. It's a market that has grown along with Anvisa. And in order to have different assessments, we need to treat people differently, but with some isonomy that the law requires. Thank you. We have one more question, and then I have a comment to make. Good morning, everyone. My name is Elisandra, and I'm representing Abipina here. So I'd like to congratulate Jack Leckerman for the event, and especially Dr. Freitas, who is giving us so much information, and also talking about, the biosimilars market and expanding access for our patients. So it's very interesting to see the interest that Anvisa has in doing that. So you mentioned that one of the biggest challenges is about proving efficacy and safety, clinical trial conditions, preclinical trials. Brazil is now better at that. But I'd like to ask about clinical trials. In a national lab, if you have 100% national development, there are still some challenges in the regulatory requirements for clinical proof. So in this initiative, with this public notice in raising the main challenges in Brazil for this development, do you intend to also have that specific outlook to attempt to speed up the process of approval, especially for the conditions of clinical trials? There's an additional step that we have here. Of course, it's very important for development, but it becomes a relevant point, and it even makes companies to try to develop products outside of Brazil to make their approvals faster, so that products can be made available faster as well. So it doesn't take that so many years. So is there a concern on that about clinical approvals? Is that also something that is being discussed? Thank you, Lisandra. So I started by saying that we need to be an agency for access, and in order to foster access, we need to consider the product's life cycle. We had a new regulation in Brazil for clinical research, and I participated in it, as you know, RDC 9. And we know how our market was still immature for clinical trials. So that's why we didn't use that strategy to use assessments from other authorities, so that we could have a fast track for approval. So these were dossiers that Anvisa needed to analyze, because Brazil was changing that scenario in Brazil by using DDCMs, a clinical dossier. This was a new regulation. However, we can say that we have advanced significantly. This industry has matured. Clinical trials have matured in Brazil, and even sponsors. So I think that for certain products, not all of them, but for certain products, Brazil needs to broaden its access. And we're talking about high-cost products, products that impact the entire chain directly, especially for diseases that are becoming more chronic, and that will lead to public health losses and expenses. So we need to consider the country's needs for approval, and that's also on our radar for weighting our decisions. We need to consider everything, but if we have a very new company, a new RPC, we need to look at the researcher's grandfather's DNA, as we say. But we've made huge advances in that. You can consider different product characteristics. Thank you. We have one more question here. Good morning, doctor. My name is Marcelo Paiva. It's a pleasure to meet you in person after so many interactions online with COVID. So we'd like to reiterate that we recognize Anvisa's leadership there, represented by you. Congratulations. I think Anvisa did a very good job in the last few years. But my point is about the responsibility of Anvisa's scope. As you said, Anvisa should be an agency for access, and it also influences prices. So I'd like to hear from you about two things. First, how much do you think prices have influenced that statistic of having fewer products? And secondly, we know that there has been a recent public hearing discussing that with the government. I think Anvisa will review biosimilars pricing policies. And how optimistic are you about that? Because we have that proposal from Realize, and prices are still a bottleneck. So I'd just like to hear your opinion on those two things. Thank you. Thank you, Marcelo. And I also have to acknowledge Marcelo, because we had that issue in Brazil when there was a shortage of anesthesia and sedatives, and companies were invited to increase their production. And I counted on your help with that, so thank you. Considering prices, from what I've seen, it's a global issue. It's not specific to Brazil for biosimilars. We know that there's a variation according to each molecule, but what we have discussed in approval, trying to work with several secretariats, is understanding what are the challenges in developing a new drug, including biosimilars, because they require investments. There are different biosimilars. Some of them have smaller molecules or larger molecules, and companies need to have good investments in their production process and in studies. You cannot look at biosimilars just as you look at generic drugs, which are already established in the law as having a lower price. Knowing that there's a lower number of biosimilars in Brazil also means that they will come at a reduced cost for the public healthcare system. So there is no rule for biosimilars that requires standardization. You need to have a good guideline, but you need to consider each product. And this is what we in the approval area have been trying to create awareness for secretariats. But the Ministry of Health is also need to be aware of that, as they are the major providers of public policies in Brazil.
Thank you very much. Once again, a round of applause for Merusa representing the agency Anvisa. Thank you.
Next guest that I just like to invite is Dr. Daniel Freire. He has 15 years of experience on the pharmaceutical market. It's important to note that we just started to talk about regulation, we talk about price, and now we heard about access. So Daniel has a long experience in the last 3 years working for Sandoz as Regional Medical Affairs Director. So, Daniel, bem-vindo.
Muito obrigado. Agradeço o convite para participar desse.
Thank you very much. I thank the invitation for taking part in this discussion. Well, the idea is talking a little bit about barriers for biosimilars, but also in the part of education, concepts for biosimilars that, as I will show you, there is a major gap that somehow prevent the acceptance of this type of medication for the different stakeholders, by the different stakeholders. And I will also address immunogenicity, that although it is a fundamental issue of evaluation for any drug, for biosimilars, oftentimes there is no deep understanding of how the immunogenicity occur for a biological and what is the implication for biosimilars. And finally, a little bit about opportunities. Well, this slide is from a study published in 2014, and where the authors evaluated the possible barriers for entering the Belgian market. And they evaluated three major barriers. One, regarding healthcare professionals, understand the biosimilar concepts and the importance of switching once we consider that for the market to fully use biosimilars, it is key to have a switch, because considering that just naive patients can use biosimilars, this limits a lot the impact of biosimilars. Another barrier where we should work is regarding patients. Guarantee that they will have efficacy and safety as they have with reference drug, and avoid the nocebo effect. And for payers and hospitals, establish clear policies, biosimilar first for these institutions, and reimbursement policies. When we observe the two main or the two first barriers I mentioned regarding concepts, we see that everywhere in the world, not only Latin America, but everywhere, there is a major gap in knowledge regarding biosimilars. Regarding doctors in Latin America, and what we observe is that 30% do not regard the extrapolation. 54% believed that the regulatory approval pathways for biosimilar was the same as innovative biologics, and just 50% believed that clinical experience will be equivalent between biosimilar and reference drug. Well, a recent systematic review of the literature confirms this lack of awareness about biosimilars, or lack of knowledge. In this chart, when healthcare or healthcare professionals were asked about their knowledge on biosimilars, 75% said they were very familiar, but when we measured this knowledge, up to 86% believed biosimilars and generic were the same, and 75% informed identical structure between biosimilar and reference drug, confirming this gap of knowledge about biosimilars. Well, basically, this gap is due to a need of paradigm shift when we consider a biosimilar in comparison to reference medicine. In a reference drug, we have a clinical centric view. We have to prove by clinical assays that this drug is safe for the applications. Biosimilar is just the other way around. So here is not clinical centric, but proving the similarity between molecules. So the base of the exercise here is analytical. Clinical essays are just to confirm. And we as clinicians, for us, understand and accept that. Some other conceptual challenges for biosimilars. First, the variability of a biological, especially if we talk about therapeutic glycoproteins. Doctors will never thought by the industry that there is a variability between lots. No lot is identical to the other. And therefore, we cannot imagine absolute identity between biosimilars and reference drug. Another concept is a perception of abbreviated approval pathway. Well, it is abbreviated in terms of clinical assays, but it's as rigorous as. So abbreviated can be noticed by, can be understood by healthcare professionals as less rigorous. The totality of evidence, having clinical assay in a center of reference biological and go to physical, chemical, and analysis, which is more important when we compare molecules.
Passam a ter exatamente papel confirmatório e, mais do que isso, o tipo de estudo clínico é diferente, os endpoints são diferentes. Quer dizer, para estudo em oncologia, pensar em...
Type of clinical data. In oncology, overall response rate for a reference is maybe not that important, but for a biosimilar is more sensitive.
Juntando a esses desafios conceituais, desde os começo dos anos 2010.
And together with that, since 2010, we started to see alternative sources of information with a major amount of misinformation about biosimilars. So what I'm trying to say is that in addition to a paradigm shift for most healthcare professionals, there is a misinformation line about biosimilars that are on the reach of a hand of most professionals, which has had an impact for these drugs. I will mention examples. This was read in current EU pharmacovigilance is inadequate and cannot be extrapolated for other markets. This is not true. This is fake news. This is misinformation. EU pharmacovigilance is really robust and has 2 billion patients treated with biosimilars, more than 90 products approved without withdrawing from the market due to safety issues. Correct information, but out of context. We may say that there is no infliximab that is interchangeable and approved, biosimilar approved in US. But if you consider interchangeability of FDA, this concept is only valid for products distributed on community or for drugstores, but not for hospitals. So this affirmation makes no sense. Incomplete information. Biosimilar are not identical to the reference biologic. Well, there is difference, but these differences are absolutely controlled from the standpoint of safety and do not represent any experience difference for patients. And a totally fake affirmation. There is still a controversy for safety and efficacy of biosimilars. This is not true. This is already defined and asserted by the experience in the market. And finally, a negative framing of terminology, non-medical switching. This has a huge impact for patients. Then saying switching to a biosimilar, non-medical switching sounds like the doctors are against that, physicians are against that. And actually, we are talking about exchanging for equivalent products. Now I will address a topic that is very important. What is about immunogenicity in a biological product, in a biodrug? How does this occur? How do a product lead to immunologic response in the body? Well, this is the evaluation of immunogenicity is very important because any theoretical problem that might arise is immunogenic, especially with complex proteins. And the formation of antibody, anti-drug antibody is related to the to reduction of the drug activity, increase of the effect of the disease. But how does anti-drug antibodies are formed? Well, there are two main pathways, T-cell dependent and T-cell independent pathway. In the first pathway, a cell presenting antigens captures the antibody or the drug we are supplying, degrade the molecule in linear peptides and present the fragment of linear peptides to a T helper cell that interact with a B cell to produce anti-drug antibodies. This is the T cell dependent pathway. But there is another pathway independent of T cell where an aggregate of this product, like an antibody, for instance, monoclonal antibody, will interact directly with the receptors on the B cell membrane, producing anti-drug antibodies. What is the importance of that for biosimilar? Well, first, considering the first mechanism, T-cell dependent. As I said, the antigen presenting cell will break in small linear peptide segments. The amino acid sequence of a peptide is the same as the reference. So the immunodominant epitopes for these peptides are exactly the same, are identical. So we wouldn't expect that a biosimilar, although it has differences in glycolization process or post-transductional process, we wouldn't expect any difference in this mechanism. As for the T-cell independent pathway, that depends, as we see here, of aggregates and impurities. Again, these are characteristics that are highly controlled in biosimilar production. So, biosimilars may have less aggregates and less impurities compared to the reference, but never to have more. So we wouldn't expect that through this pathway there is a difference in terms of risk of creating anti-drug antibodies for a reference drug. But this is not the only thing we have to evaluate. Many of the infusional reactions for injectable drugs are not mediated by anti-drug antibodies. Some of them are mediated, but not by these processes I just presented. There is, for instance, immunoglobulin E, IgE, that may lead to anaphylaxis. So I will talk a little bit about anaphylaxis for immunobiologic, and I will use as example cetuximab. This is a monoclonal antibody against IGF receptor used for colon carcinoma and other epithelial carcinomas. And this drug has a risk of true anaphylactic reaction during infusion. The level or the rate of reactions is 3%. And we observed that some regions in US, the incidence of real anaphylaxis increased to 22% of the cases of the infusions. And we identified that the mechanism of the anaphylaxis to cetuximab is due to the presence of a non-human sugar. It's a chimeric antibody, so there's a human part, and the experimental model, the antibody forms or allows the presence of the glucans, that is galactose alpha 1,3 galactose. We humans do not have enzymes to reduce this saccharide. So there is a possibility of forming an antibody against this sugar that we cannot digest. So it is interesting that we proved that the regions where there was a high prevalence to anaphylaxis to cetuximab is the same environment where we have endemically endemic ticks. And when the person had the ticks, this increased the antibodies against galactose alpha 1,3 galactose. They already have the preform antibodies, and they have the reaction to the first presentation that was actually not the first, because they have already been presented by the tick. And one of the most important informations is the glycosylation profile of a biosimilar compared to the reference. And it is mandatory, although it is not identical. There is a variability, even in the reference from batch to batch. The major glycosylation has to be similar. For instance, comparing rituximab reference and two biosimilars, the same 25 glycoforms were identified in the products, and non-immunogenic ones with the galactose alpha 1,3 galactose. But there are, as I said, other mechanisms for infusion reaction that are not immunologic or anaphylactic. For instance, the cytokine release syndrome. What is that? It is a clinical disorder characterized by symptoms related to anti-inflammatory mediators storm right after the infusion. There are 4 mechanisms to which we can attribute, and oftentimes there is an overlap to release cytokines. The first, direct connection of the biodrug to the effector cell. For instance, here, rituximab connecting to CD20 in the surface of B cells, and this may be observed in a patient with a high number of B cells, like a patient with lymphoma or leukemia. And what is important for biosimilars? The effector biological function has to be equivalent. So biosimilar, the capacity of binding to the CD20 and activate this site, there is a variability, but this variability is within the range of reference medicine. And therefore, we cannot expect a different response in a biosimilar. The other mechanism involves the binding of effector cells with non-effector cells through the Fc receptor on a non-effector cell. So is the other part of the monoclonal antibody that we are talking about. For instance, we may have example 2 to the left. The union of B cell with a macrophage through a monoclonal antibody, and B cell will release more cytokines, or the non-effector cell may release, as the example to the right with a macrophage. All of those depend on the binding of effector and non-effector cell through FC receptors. And this is highly controlled. Here we have the coefficients binding coefficients for reference and biosimilar. So there is a variability, but it is always correspondent to the reference drug. And finally, it may exist an anti-IgG antibody formed against the biological, and once this biological is interacting with the effector cell, the antibody binds and releases cytokines, and this triggers cytokine release. But again, there is such a high similarity between biosimilar and reference that the studies evaluating cross-reactivity show that reactivity against reference and biosimilar is exactly the same. In other words, the system, the biological system and reference with the same molecule, and there is no there's no reason for being different. And there's a lot of literature about switching studies, and both real life and clinical assays. And the most recent meta-analysis published in 2020 evaluated 20,000 patients that went through a switch in 178 studies with 10 molecules. They were mostly real-life studies. And what do we observe? So far, the analysis conclusion doesn't show that the reference for a biosimilar leads to efficacy problems, safety problems, or immunogenicity issues for the patient. And more recently, we published another systematic review in biodrugs on studies evaluating switch from biosimilar to biosimilar, which is another topic of concern.
What this meta-analysis showed, there were 23 studies over 1,500 patients analyzed with 4 molecules, infliximab, adalimumab, etanercept, and rituximab, and anti-TNF antibodies where there were higher concerns. They are immunogenic, and all of these studies are observational. The conclusion is that no efficacy reduction was observed or increase in adverse events were detected in switching between biosimilars. Finally, I think it's very important to be aware of the importance of information on what I have mentioned, this gap on biosimilars. Because if not, then we're going to have an ominous cycle of adopting the original drug, which can even reduce the adoption of biosimilars, which is negative considering the opportunities they offer. If a healthcare professional does not know enough about biosimilars, this will generate a higher nocebo effect. It will generate a worse clinical experience. This experience is going to lead to higher health expenses, switches to more expensive drugs. It will get in the way of the sustainability of the healthcare system, and obviously, with fewer resources, access to biologicals will be lower. So therapeutical recommendations will need to consider them in a more advanced stage of the disease, which all will lead to higher expenses, lower health in communities, and misspending when you have limited resources. Finally, to conclude, the first message that I'd like to leave is that disinformation on biosimilars needs to be considered. This knowledge gap cannot be ignored, and we need to speak out when you see it. There are many entities that are open to receiving reports on misinformation on biosimilars, whether they are campaigns from the pharmaceutical industry or even publications in journals. The FDA, the US FTC, and ICMRA are open to that. I think Anvisa is as well through its whistleblowing channels. So this needs to be discussed as much as possible. It's important not to forget about it, and reviewers in scientific journals need to be aware of it. I've seen journals discussing risks of biosimilars, but when you look at the articles, they're not actually talking about biosimilars, but biocopies of products that have been approved in markets that are not regulated with no biocomparability studies. So they are not true biosimilars. Another topic for discussion should be teaching about biosimilars in nursing and pharmacy programs and all medical programs. And finally, patients should also receive information about biosimilars. It should be accessible information in a graphical format that will allow them to understand it better. So I think that's what I had to share. Thank you again. And I'm now open for any questions. Thank you, Danielle. I think that was a very clear talk. I liked the summary, especially when you said that there's still a long way to go to encourage universities to discuss biosimilars and include it as a part of their curriculum. So it's a new topic, but it does need to be explored. So shall we open up for questions? Do we have any questions? There's one at the back, and after the coffee break, we're going to have a raffle. And if you're sitting up front, you'll have a higher likelihood of winning. So you should move up here. Hello, good morning. My name is Tatiana San Juan, and I'm from São Longuinhos Fiocruz. Thank you, Danielle, for such clear presentations. Thank you for this partnership. When you talk about clinical trials, I'd just like to understand what time you considered to switch, and if you considered multiple switches. So that's an important point, especially in the public healthcare system. We see covert movement from the pharma industry against these markets, and we are very concerned about how it's been done and how we can support this strategy. Thank you. Thank you, Tatiana. So the time varies, but what I can say is that the minimum period to maintain a product is around 6 months. There's another discussion here that we should have on how much it makes sense, because I agree, multiple switches often raise many concerns, but what makes sense, even from the healthcare system perspective, is to allow multiple switches. I mean, multiple switches are a problem in bureaucracy. First, you have to deal with that error in bureaucracy because there are no advantages in having multiple switches. But it means that there were some failures along the way in implementing that product for that population. But switching after, let's say, a year or 2 years, which was what was observed in most of these studies, after a minimum time of 6 months, did not show any issues. First, I think we need to consider what we want to do, and then based on that and based on data from literature, we would be able to do it. But I don't see much sense in finding evidence for switching every 3 weeks to see if it's safe, because it doesn't make sense to do it. Great. Thank you, Danielle, once again. We're going to continue with the next session. I don't think we have any additional questions, so we're going to continue to the next session. Once again, thank you, Danielle. Thank you. Well, it's a pleasure to introduce Matias Fassolari from Buenos Aires, Argentina, who has joined us. He has a lot of experience at Merck, and I think is he going to speak English or Spanish? Spanish. Okay, so now we're going to hear a little bit from him. Welcome, Matias. Thank you. Okay, thank you. My name is Matias, and I'll speak Spanish because I think you'll understand me better when I speak Spanish than if I were to speak Portuguese. I'll try to speak slow. And today, we're going to talk a bit about next generation bioprocessing. It will be a little bit more technical, so I hope that you enjoy it. We talked a little bit about a paradigm shift in producing biological drugs, including biosimilars. So let's talk a bit about what this paradigm shift is from the production of the drugs and what is on the way in the industry. Then we'll talk about how some small changes are taking place for the entire process and how that will lead to improvements in the upstream process. So first, let's talk about why we are discussing process intensification. As you know, there now is more pressure to develop production processes, and the pharma industry is focusing more and more on that, as well as filing new patents and releasing and approving more biosimilars in the market. As we see in industry leaders, there need to be some paradigm shifts to become more efficient. And this has been discussed for a long time, that the industry needs to follow the trends we see in the entire market, among which are a higher speed of production, faster time to market, having better processes with lower costs and more flexibility. That is reducing cost as much as we can. And, with flexibility, it would be having a production line, being able to produce several molecules. And of course, in biosimilars, there's a high cost pressure. So for that, we are developing several technologies for production, which are listed here in the last row: process intensification, process analytics for what you are manufacturing, and a more connected and automated process is also important. So in this new generation processes, what are we seeing in the industry? Basically 5 things: increasing productivity, increasing flexibility, as I said before, having more production areas, but with lower production time, and also producing at a lower cost. But how do we achieve that? That is a good question. How do we reach that by having a smaller factory that is more efficient? So there was a survey with 30 big biopharma companies around the world, especially in the US, about how important it was to include next generation processing, and what were the benefits that they expected to have by using it. And the figures show the most important drivers. 93% wish to have greater facility flexibility, better cost control, and 77% wanted a more simple process, using the same production lines and having a more robust and reliable process. So these are the main concerns for the industry today. And for the pharma industry to continue with next generation processing, many disciplines and many technologies need to advance. The process, well, here we're seeing where we are and where we want to be. As you can see, some of them have been advancing. For example, controls currently are more intensive, and they allow us to reduce production times and have a higher level of production, but maintaining the same qualities and the same scale. The aim, though, is to reach a totally continuous process that is connected across all units. The format as well. Today, we're using a stainless format, but we expect to take it all to single use so that we can become more flexible and closed overall. When it comes to analytics, we are currently using standard quality control methods. But by using better equipments, you can have continuous analytics. Of course, it is more technologically demanding, but with time, you can have real-time sensing that would increase product quality. And that would not remove much from the process, and it would actually increase your real-time reactivity. Controls, as I've said before, are going from a standalone control method where you can have a more predictive one, returning to your analysis a more complete perspective on what the process can change. And each process has its own control now. Instead of being centralized, there's one person controlling each process, but the idea is to be more connected and grouping different levels of control together, leading to a fully processed control that can be automatic and, if possible, in the future, autonomic as well.
These are the productive processes we use currently, which are each circle you see as a standard process where we have the production of cells to produce. Then we have a stage of purification and elimination of possible contaminants. And then the packaging. We have to group as much as possible all these units and improve the process, decreasing times. And in some years, this infrastructure will be a continuous and compact process. In order to do so, we are working in different intensification platforms that we will address later on. The trends for molecule production and the evolution for a biosimilar, and all the stages of purification, and finally achieving a product ready to be packaged and distributed. And everything has to be followed by analytics and a better control of the process with less variation. And this generate data to have a predictable process, to know the variabilities and avoid dramatic variations in the process. That will, in the end of the day, impact the quality of the product. So upstream is cell cultivation. Here is summarized. So basically it's an upstream process. Initially, we have cryopreserved cells in a working bank, in a typical bank, and we amplify the number of cells through consecutive culture, generating amount enough to go to a bioreactor and produce in scale. This is marked here with N. From the productive standpoint, this process is not patent and very efficient. And efficient meaning that we are very much concentrated in the initial phase, which is the growth of biomass of the cells. Here we do not have the product itself. But we need an amount of cells, a mass of cells enough so we can start producing. How can we improve this? How can we reduce initial times to have a better use of time and dedicate more time to the production itself is part of what I will show you right now. So, how can we intensify the upstream? What we will see here, basically we can do it in two different phases. What we call seed train, which is the expansion of the cells through different mechanisms. One of them is high cell density cryopreservation, HCDC, and the perfusion, which is having a reactor before the productive reactor. This can be intensified through continuous culture of the material, the biological material. But in the seed train allows for intensifying the production in a simpler fashion that can be carried out without much change in the productive process. So we intensify the seed train. There are two mechanisms that I showed you in the previous slide to obtain HCDCs, which are 10 million cells. These cells are cultured in bags in this case, or tubes, but bags in this case, leading to 50 million cells per milliliter in 100 milliliters culture. And the benefit we have here that we can eliminate one of the loops of the process, decreasing the time for expansion and perfusion of the cells. And another important part here is that the initial phases are opened operations. So the culture is exposed to possible contamination. So in closed bags, they are enclosed from beginning to end. So it is better with the ACDCs. And the other option is increasing the perfusion to the bioreactor in the end part, in the downstream. The benefit here is that in the perfusion, the cell density in the culture is much higher, 10 times higher than the normal. So we can improve the culture to feed the final bioreactor. And again, we can eliminate one of the phases with bioreactor. So, high cell density cryopreservation, as I mentioned before, is a technology that is currently developed. This is the assembly here, the array, and is a completely closed loop. It is already proved that is the best way to handle it. And the other benefit I mentioned before is that allow to decouple cell expansion from production and decouple the times and spaces. You can have expansion in a plant and take the system to another area or another plant for production itself. But from the perspective of production, the decrease in time is the best feature here in the cell culture. Here is the calculation of the times to achieve the bioreactor, comparing a vial with 10 million cells and a bag with 150 milliliters and 50 million cells per milliliter. And if you see in the bottom table, with a vial, it would take 24 hours to reach the final bioreactor, and for the bag, we start later on in the process, so it reduces the process to 1/3 of the time that is usually required. And in the core of the process intensification is perfusion. I don't know if it is really an interesting topic for you, but usually we have a continuous system. So we insert, we feed the bioreactor, and we have a way out and an outlet, and we maintain the cells in a culture. So here we maintain the cells, and we take out the product and pass it to a fresh medium with all the nutrients, increasing the number of cells, in addition to saving more product because there is lower residence time on the bioreactor. And the benefits of perfusion I listed here. We can compress the cell expansion phase, maximize the bioreactor operation, reduce the size and number of bioreactors required, And perfusion can also work to intensify the production, the productivity. So here what you see is the bioreactor being fed with cells and fresh medium, and the product getting out. It's important to consider the methodology to intensify the productive process, because we can decrease the size of bioreactor or the production scale, leading to a greater amount of liquid that I have to manage. An alternative, a possible alternative to intensify the process without needing perfusion is what we called feedback culture, where the cells are growing and each certain time it is led to expansion, and this culture reaches a higher expansion rate. Here I see 4 processes and the benefits. The first one is a convention batch process, where we start in a tube, a culture tube, and we pass through all the phases to get to the feed batch bioreactor, taking 14 days. The second, compressing the seed train phase, which decreases one of the phases to reach the bioreactor. This bioreactor can have 200 to 500 liters, and we can feed directly from a bag with 40 liters of culture. And we can intensify this perfusion with a hyperbatch, intensified usually the initial amount of cells in a bioreactor is 300,000 cells per milliliter. If we apply the perfusion, we achieve more biomass, and we can seed it with a greater amount of cells. We can inoculate and intensify the upstream, as I will show you later on. Next, please. So this is what we done to compare. We have a control condition, which is the conventional process, with the conventional expansion phase and a final bioreactor, inoculated with 500,000 cells per milliliter, or 5. And the second one, and this is what we've done, increasing the number of cells per milliliter and feeding the bioreactor with 5 million cells per milliliter. In the next slide, we see the conditions. I'm sorry, the sound is reaching the channel really choppy, but we are trying to do the best possible. Next, please. To set up the experiment to develop this technology for this type of process, these are chose cells, a culture medium that is called XL fusion, and the equipment to expand the cells and the bioreactor. And previous to the evaluation, we tried to optimize with the experimental conditions and adapt the cells to a perfusion culture medium. To avoid excessive perfusion in the N-1 process, and improving also the feeding strategy for the final bioreactor with 5 million cells per milliliter. And here some charts to illustrate the impact of intensification or high seed cell. Here we have the cell growth. In green is high seed intensified, and we can see I'm sorry, the sound is really choppy. So at the end, inoculated with the normal cell. We can see here that the growth peak in all conditions is similar, but the one that is intensified gets to this peak 3 days earlier. So this is what we see in the next slide, the effect of that in the amount of product. I'm sorry. With the high seed intensified process, and this is the larger growth we saw that is translated in a higher and faster productivity. If we see here picograms per cell per milliliter, we have a great intensification on this stage. So here the process with 3 days. I'm sorry, the sound we are receiving in the channels are really bad quality. So with the amount of product, what we see in the chart is the accumulation of product.
Después otra cosa que analizamos fue el metabolismo de las células salvajes.
Another thing we evaluated is the metabolic profile of the cell. So we evaluate the metabolism in different metabolites in the medium. So we evaluate to understand the growth, the glucose consumption, and the profile of consumption is very similar, producing lactate. That this is basically the profile in the intensified project goes to the left of the chart. And we can, with that, correct a little bit the process.
Finally, here we see the product quality attributes, and you can see that they have a very similar level. We analyze product degradation, and we do not see a big difference between them, as you can see in the SEC analysis. It includes aggregates and monomers. And finally, we concluded with a charge analysis. And as you can see, there's a higher acidic rate in the in the second process. So as the process advances, this is corrected. So from the perspective of the process, what I wanted to show you is that intensifying the batch increases some of its acidity. It's a process that is difficult to carry out, but it can be done through modifying some of the current processes. So these are my takeaway notes. What we say is that with Actin, there are several ways of having intensification processes. Seed train intensification with high cell density cryopreservation or perfusion at N1. And you also need to consider cell expansion time. Offer the option of higher flexibility, and it's a process that is completely safe from contamination. And perfusion N1 allows you to compress the seed train, eliminating it or making it lower or smaller, maximizing facility capacity and efficiency in producing the molecule, reducing the size and number of bioreactors that you need to obtain the cell density that you required, and it also reduces costs of manufacturing. That's also very important. There are other technologies to intensify processes that can complement what I mentioned. Increasing seed trains, intensifying the upstream process, and some of these technologies are creating a revolution and advancing and improving some of the results as I have just mentioned. So that's it from me. Thank you, and if you'd like to contact me, this is my contact information. If you'd like to get more information on process intensification, feel free to contact me. Thank you, Matias, for your talk. If you'll allow me to ask a question, this technology requires a lot of time and investments. In how much time do you think that this will be ready for biosimilars? When can we try this technology, and how much time would it take to reach that stage? Because, of course, this would all need to go through the regulators, right? So how much time do you think it would take for these results to reach us? Basically, what I wanted to show you is that there are many things on the way to create a batch intensification process, but it's very difficult because the cell lines we're using might not work with this process, and that would require a major change. So we're working on the first batches to be launched, intensified. And of course, if we can use the same cell line, it will depend on if you already have the technology or not. So between research and implementation, it can take 1 year, but then regulatory matters have a different timeline. Okay, do we have any questions? So that you have already answered my first question, but do you already have enough technology to test?
Bags. The bags.
So how do you characterize bags? It seems very interesting, but I'd like to understand if there are any practical applications of it. Okay, I'll answer that. Currently, what we have are these bags not for banks, but for processes. So our process is to create and expand and freeze in bags the regular processes and then start it for a production process. So this is what we're developing in banks. Well, the current ones that we have only support minus 80, so we're developing bags for liquid nitrogen. So space is also an issue, but this is all being developed. We're not sure what will be implemented. Our aim is to have these bags, but so far we only have the technology for them on a medium size. Okay, before we break for coffee, I'd like to summarize a couple of things.
It was 3 points that's very specific, very important, that can give insights to us. First is a regulatory pathway with Anvisa. What a great presentation. We understand the challenge that we have ahead of us for biosimilars. Also, the point of view from the medical and perceptions about biosimilar that we heard, it was great. And now some improvements on production. Matthias, before we go to our coffee break, that we last question. It's a more personal question. ¿Argentina va a ganar el mundial este año o no? No creo que sí, no creo que sí. No vamos a contestar.
Will Argentina win the World Cup? No, I don't think so, but we can talk about it over coffee, and we'll be back at 11:40. And if you can sit ahead, some of the chairs have some numbers that will be used for the raffle. No, but really, if you can sit ahead in front of the room, because we will have round tables and it will be better to interact with people. So if you're sitting at the back, if you can please move forward, and we'll be back at 11:40.
Muito bem. Acho que a gente pode voltar para nossa próxima sessão.
Okay, we can begin the next session.
Let's come back to our next session. Okay, we still have some chairs available here. With now it's a $300 with an envelope under the chair. Please move on. The price is not going to be high. So for the next session, it's a great pleasure for me to invite Tiago to be the moderator of this. Tiago is from Butantan. I have a lot of respect from Tiago. We met in some projects together, but also I should recognize that Butantan, it's an outstanding company here, public company that we have in our country. Tiago, thank you so much to be here. I think on behalf of 200 million people that we have in our country, thank you to Butantan. That's the first company that brings vaccines to COVID in our country. Thank you to be here and to be the moderator for this important session. Couple of points here. We have also participación de Leucotec México. Y también invitar a Mariana Duarte, por favor, a se reunir con nosotros acá. Esta sesión tenemos una discusión un poco más abierta con preguntas y respuestas. My understanding that we go for the discussion partially in Portuguese, partially in Spanish, partially in Portuguese, and at the end you understand what does it mean portuñol. El mix. Tiago, por favor.
Bom dia a todos. Obrigado, Marcelo, pela...
Good morning, everyone, and thank you, Marcelo, for that introduction. It's a great pleasure to hear these words, and we're returning to in-person events. So at least here in Brazil, this was due to a major effort from the public side, not only Butantan, but also Fiocruz and Biomanguinhos. So this is a legacy that will be left to our country. It's a priceless asset, and we're also going into the biosimilars front because we work with prevention, but in many situations we also need to have the production capacity for therapies. And this kind of event is very important to foster an exchange from our region to other regions around us as well. So thank you everyone for being here. And welcome to my hometown. São Paulo is my city of origin. I started working here in Paulista, and then I went to Butantan, the other side of the river, where we work with biotechnology. And we're going to be talking about single-use systems, hybrid systems that we used to see 15 years ago, and now we see so many of with the entire single-use production system. So we're going to have a very important panel with our colleagues, Martin and Mariana, who are going to discuss their experience. And then we hope to get some questions from the audience. I'll start with a question for Mariana, Women First. Mariana, as a representative of the biotechnology industry in Brazil, how do you see these new trends for production platforms for the next years? Technology development is very fast and dynamic, so how do you see it, and what trends do you expect on the technology front? Well, first of all, thank you for inviting me to be here. Thank you, Tiago, for that introduction. And now we're going to speak Rio de Janeiro Portuguese, but I am from Rio. I have worked at Paul since 2016. I started in a different era where we didn't talk about single use as much. Paul is a very well-known company in the market for its filters. And in 2016, when I joined the market, we basically received calls on filters. We didn't talk about single-use and complete plants, but Paul, through some of its acquisitions and some of its own technology development, has gone into the biotech and biological industry. And currently, as a trend, we see single-use plants because of their flexibility and integrated solutions. So we're no longer working with individual equipment units. You have to integrate the entire plant and work with biomanufacturing with your clients through an entire project. So we no longer only sell single equipment or single solutions for one stage of the process only. The big trend in the market is working with a comprehensive system and a complete plant. And even in clean lines, there are now modules. So in engineering, we also see as a big trend in the market today. Yes, so we've moved along since the first discussions 15 or 20 years ago about reusing filters instead of using them. For silicon tubes first, and then replacing some items with bags. So in 15 years, we started using single-use plants. It's a very fast tech innovation, and it's great to hear from your perspective because, of course, it matches what you see in your company and in the entire market. Thank you. Martin, let me ask you a question. So Martin is going to tell us about the front he is working in. So he is more on the distribution and service to market side of things. He'll talk about his company, but the technologies you see, how do they contribute to developing the products that you work with? If you understand my Portuguese.
The elements I use for reproduction are related to single-use technology. We have a production line and 3 different products where I use different processes with the same vendor, which for me helps a lot, especially in the revision of costs, because I use the same single-use system for different types of products. They pass through the same line. So what we do, it is vertification and unification of proteins and purification of proteins. And we are using this single-use technology, which enables a better processing and a better productive scheme for the purification of proteins that enables reducing costs. Because the single-use prevent cleaning and storage of containers mainly. And can you explain some of the technologies that you have used successfully in the production line, and even the rationale for the changes? We have examples of changes of materials that we felt safe regarding cross-contamination, the contamination of the material itself, the risk for the operator. We saw some accidents in the past with glass vials, and so it goes beyond financial issue. So if you can explain a little bit about the exchanges or industrial application of these components. Well, regarding the use of glassware, we no longer use glassware since the beginning, because we are using, in the case of glassware, they lead to problems in storing them. Also for cleaning and sanitation. With a single use, we reduce time, we reduce cost with storage, we reduce man hours for cleaning, for sterilizing, and so cost and time. And because these single-use systems, for instance, filters and bags, where we store all this, the protein filtration, this enables a greater level of flexibility on the process. So we are faster without having to store, which I insist, storage is an important problem, because the more containers you have for this type of elements, the bigger your warehouse, and you need a special warehouse to store them. And in this industry where I have my production, my objective is not storage, and my main objective is producing something of high quality with cost efficiency and really high quality for our products. Thank you.
So this is a question I will ask to both of you, but let's start with Mariana. Specifically for biosimilars, how do you see, for whoever hasn't implemented them or is building a factory, revamping a plant, what improvements can you see specifically for biosimilars? Especially from the experience that you've had abroad that you can bring to Latin America. What do you think is applicable for biosimilars? Do you mean partnerships? I mean overall, the use of technology for biosimilars. What improvements can add value for manufacturers? I think it's working. So, the use of single-use systems for biosimilars bring flexibility. So currently, with a plant that produces one kind of molecule, if you need to include another molecule to the same plant, the transition is much easier. You can simply opt to change procedure parameters or simply the manifolds, or you can switch from one equipment to the other. The transition is much more simple than traditional stainless steel plants where you need to build a new area for that, which will create time and cost, and the innovations we see in the single-use systems in that sense help us to accelerate time to market for the different molecules. And also, you will need less space. So in smaller spaces, you can use the same technologies developed for the same area, so that will allow for more productivity. And that will also be a gain in using the new technologies. And Martin, about what you said, specifically for biosimilars, what improvements do you think are possible for production in implementing single-use systems?
Tratando de entender el portugués. Las mejores que ganas en la producción de biosimilares con el uso de sistemas de uso único.
Pues, y como mencioné, y parte de lo que había platicado hace un momento, lo que me faltó comentar es que el uso
What I like to comment is that the use of single-use technologies reduce the risks that may occur with cross-contamination and reduce the number of containers, reducing the risk of contamination in having to recall a whole product batch. So it is single use, you use it and go to the next one. And it is very easy to maintain a traceability. So when I release a batch, it is safe and high quality. And at the same time, I can have different productive lines working, and which is much simpler. Instead of having a huge container that you have to clean, sanitize, this is much faster. It is this technology is really flexible and allow doing many things. For instance, we have process of different products right now, and this type of technology works for the three of them. So it is very agile.
Just to add to that on contamination, besides batches, the financial issue is important. We know that biologicals give patients a lot of quality of life. So not providing that to them will affect patients directly. So it's also very important. And single-use technologies will reduce the risk of contamination and prevent against losing batches. Yes, without a doubt. And also as a manufacturer, the plant will have a risk and a big maintenance effort if it does not do that. And that can lead to higher downtimes. It can impact the distribution of products. So this is definitely a factor that will improve a manufacturer's daily life. If you're developing a plant, if you're on the conceptual phase of a plant, and here I'll mention my own experience. This system is the reason why we're putting it into context. It facilitates the development of new products. We heard about Miruzi's talk, how easy it is to launch new products, but it's also important to bring that to patients. So replacing products in a production line provides that flexibility, and therefore it is essential. So a biosimilar plant, I know it's a very specific market, but when you look at different regions such as Brazil and Latin American countries, that can be an important step forward. So being dynamic in replacing products is essential to use this sort of system. Specifically on the best control for using the system. We've talked about cross-contamination, biosafety, but to understand the use of a system, especially in direct use biosimilars. So what can you say about controlling the material use? Yeah, I can approach that in a different way. So before COVID, we had a scenario where materials were delivered in a way, and after COVID, we had some issues with raw materials, with components, which affected our delivery times. In a conventional plant, the partnership between the supplier and a consumer goes one way. But here, it's different. It's shared responsibility. When we talk about a single-use plant, you need to have better levels of controls for your inventories. So you need to have a partnership with your client, and it needs to be much more significant to ensure that there's no rupture due to the lack of these consumables. And during COVID, we faced many issues. With that, we have issues related to the component. So now we have a single-use production line, and not all of them are in Paul. Some of them are third parties and are included in our production line. And this is brought to our responsibility. So what we've seen is that companies have massively invested in expanding capacity and are also starting to produce some of these components that before depended on delivery times, and now we're incorporating them to our own production. So we've been producing some of these components as well. So when it comes to deliveries, you have to see controls as a shared responsibility between the client and the manufacturer, the supplier. And besides that, we also have some regulations, quality. It all needs to be notified. Changes in components need to be traceable. So the entire quality documentation needs to be up to date for validation and controls. So in keeping the process running without any downtime, that's what it can add. We see that there has been massive investment in increasing capacity. And we're also attempting to minimize any dependence on delivery times from others. And internal controls, we also have closed systems, so that prevents contamination, cross-contamination as well. You can use a bag that can simply be replaced instead of needing to be washed in a tank and validate its cleaning. So in the single-use system, we also use some processes to control. In single use, it's validated, it's ready for use, so it also generates more control. In complete plants, you also have easier automation, so that also gives you more control and a better overview of the process. So these are a couple of things that I wanted to mention. There's control where you're not going to stop producing, but there's also control for the production itself. You mentioned a very interesting thing. I think that as a concept for the industry, for manufacturers of biosimilars, if you have a plant with a single-use system that is already implemented, you have a higher relationship than simply being a provider of a material. You become a part of the production system. So there's no way of dissociating that responsibility in the production chain. It's a part of it. Without it, you cannot get an easy replacement. So this kind of responsibility for suppliers, and now I believe that they're a part of the production chain. They're not simply providing a consumable, but the responsibility that they have is also very important. And we've heard that from our partners as well. Martin, do you have any experience with controlling the supply of materials in your production routines?
Well, Mariana explained it very well. In controls are completely different in a single use than in a normal plant, in a conventional plant. Controls are easier because you validate and implement single-use protocols. You reduce hours and labor. The qualification of the areas and the whole system, actually, by reducing that, you can be more efficient. And this equipment and this technology are elements that come sterile from factory. So it saves time because you don't have to sterilize and sanitize again. So you need less controls, and with that you can be more efficient. I also remember my experience working in a productive plant. The management, the material management that had to be prepared to be used because they were not ready to use, and that was really complicated on from the production standpoint, because we had guaranteed cleanliness, sterility time that this sterile material was stored, increasing complexity to manage the plant and requiring a more effective control. Do you agree? Yes, I fully agree. With that, you reduce mainly the most important thing. You reduce the risk of contamination, which is key in the process we have with this single-use technologies. It's exactly avoiding this step that may lead to losses and these costs. If you have a cross-contamination for any element in a single that is not a single use, it represents money. You have to throw it away, you have to dispose it and start from scratch. With this process, with this type of single-use element, you reduce that.
So now we'll talk a bit about how the world has changed after COVID. Well, I shouldn't say after COVID because the pandemic is still ongoing, but there have been some changes, not only in the industry, regardless of being, active ingredients or vaccines or biosimilars. We've realized how dependent Brazil is on Latin America in importing some of these materials and even some medications. So there has been a major appeal from sanitary agencies such as PAHO and WHO to reduce interdependence, so producing biosimilars locally. So now I want to hear from both sides because we have a supplier and a buyer here. What changes have you seen in this new area of regionalizing production? Well, I mentioned this during the previous question, but since we work with single-use systems, without them, there is no production. Differently from a stainless steel company, without them, you cannot produce because the tanks and bags are provided by the supplier. So along with the client, you need to manage inventory for the inputs. It needs to be face-to-face. In our experience, we have worked with Biomanguinhos to produce the COVID vaccine. That was a tech transfer provided by AstraZeneca. So we have weekly meetings on delivering inputs to make sure that there's no stop to the supply. And some components we used to depend on third parties before in our systems, but now we are able to increase the number of suppliers that have been approved besides our own manufacturing. We now work with a software that has been developed by Paul to manage this design, and any changes made can be traceable in the software. All the documents related to quality, validation, all of it is also stored in the software, and it can be accessed by the client directly in our regulatory website. So clients can register and get validated easily. So, we cannot wait for too long to do this kind of replacement. If lead times are poor, then these systems need to be improved. And with easy access, then you can do this faster digitally. Are there any plans to regionalize these systems? Not at all, not yet. But we did expand our plants. We're expanding our own plants, and we have an additional plant being brought into the line, one in Duncan and one in China for the Asian market. So we have a few expansions there. Great. Some of our colleagues who are manufacturers saw some issues during COVID, some in replacing suppliers, but also some geopolitical issues, that chaos. Our country was impacted by some suppliers from the US and India as well. For vaccines and for biosimilars, they are one of the biggest producers of biosimilars in the world, and they were impacted by a shortage of materials. So imagine having the best production capacity, but not having enough supplies. So this has been driving regionalization. And Martin, what is the experience in Mexico and in your company? How do you see regionalization for local production?
Well, in terms of local options, you mean the technology or the products themselves? Both, because one depends on the other. Having it in a local productive system, producing technology, reduce our costs. For instance, import cost, foreign exchange, because in Mexico we import in dollars, and these will have an impact according on the type of on the foreign exchange, and really and this avoid also times expecting for the imports to arrive, and also talking about the cost during the COVID and post-COVID, we had production problems because there was not material enough, not raw material that is come from a producer that is not regional, is located in another country, and this lack of supplies affected the productive chain downstream. If there is no supply chain, if there is no raw material, we don't have the final product. So we had to change for single use, and also for the product. We have lack of product, we have lack of plastics that are used in different technologies in our inputs. So it really was a problem in our whole productive chain in terms of time to deliver. And we had to seek solutions and technologies that don't impact the final quality of the product, because sometimes you can use a substitute, but we have to ponder if this comply with this, the substitute comply with the sanitary regulations, and is the same quality of what you were using before, because at the end of the day, it doesn't work, or at least it doesn't work in the same way. Again, you have to implement new controls, and the product at the end of the day is not the same. And this affected us for not having a local producer for this type of technology. And the time to respond to market took months, but the inputs that we use for our products, or the feedstock for our products, was affected. So it was really a chaos. Even the post-pandemics, it's better to have a local producer, a local supplier. This makes, well, I think I took too long, but
So I gave the standpoint of the supplier, but hearing your standpoint from manufacturers of biosimilars, now companies can also help in regionalizing biosimilars through development services. We can now offer to our clients development services. It can be for the entire process, or it can be in stages in the process, and a bottleneck. And we can also offer to our clients these kinds of services, removing molecules from universities and taking them to clinical tests. Yes, that's a great regional support, especially in Latin America, that has developed, and can choose what system to use. So if manufacturers have that system available to them, that it can help them make a better decision. Thank you. Now, let's continue with the next question related to COVID. I think the pandemic has brought a lot of information to society about products. We hadn't discussed this before. We wouldn't discuss this before, but about cells and viruses, and what conservatives are used in vaccines, but this has created a much healthy discussion, although it generates that problem of fake news, but it provided a lot of information for society on the products that were being used. And it includes the end consumer now. Someone who's going to make a decision on what will be taken. So we've had that perception with some devices specifically. Primary packaging, an injectable versus an intranasal product. So the patient can now select what sort of administration method they like, and they can talk to their physicians about that. Considering single-use systems that the industry has already been using, there are benefits, there are some issues in safety. Do you believe it can be a factor for the decision from the patient, who will know that it will be made from single-use system, or for the physician who is going to prescribe it to the patient? So will patients start analyzing it? Will that figure into their decision process? That's an interesting question, Tiago. Do you remember what it is? No, but what you said is interesting. Before COVID, we wouldn't hear this sort of conversation. Oh, my child needs to take the vaccine. I mean, regardless of what vaccine it was, they would give it. And then with COVID, we are having this sort of discussion. What sort of vaccine is it? Is it a cell carrier? Is it an RNA vaccine? This sort of vaccine didn't exist before, and now it does. And I do believe that it can also move to a discussion about, this manufacturing plant is single use. So it gives me more safety, it gives me a better level of control, and therefore it's a product that I will choose to use. That's interesting. It could move in that direction. Yes, I believe so. So out of the platforms that are currently used, this is going to make a big difference. Biosimilars produced in bacterial cells or mammal cells. So at the level of the discussion we have with society, sometimes people will ask that. And of course, here I'm referring to the medical society or managers who create public policies and who are going to replace products in a system. But Martin, how do you see this in Mexico?
Well, actually, I believe that the final decision of a person to acquire or to purchase biosimilars will depend on three main factors: regulatory, availability of the drug, and the cost. So in terms of information, if the productive plant has a good quality based on the systems used, this will lead to a very good biological product. And the other thing, if the systems or this technology enables more availability in the market because it's a faster process, that with that we can produce more and have more medication in the market. And in the last point, if you passed through all of that, the objective of this technology is reducing costs. The final cost, that is what the user is seeking, is access to these drugs. So the end user, to the end user to know if a single use or conventional system was used, I don't think it has an influence in decision-making process. But the other three factors, the regulatory, availability of the drug, and low cost. Thank you, Martin, for sharing that experience. It's good to see that this perception is integrated in the region, and it is a very interesting point to observe the level of awareness of society of what we do, we manufacturers, partners. And COVID brought this vision of what goes on in a biological product plant. So every day we saw on TV an area where a product was being made. We never had been exposed on TV before. Never had the society didn't have this information on how a biological is produced. Now we see the person that is dressed in uniform because it is in a clean area, or because it's a biological product. So discussions in the society started to happen, discussions that we didn't have before. Usually we had plants with car assembly, but never on how a biological product was made. This is more evident when we have a local system, and the TV the channels were playing that all the time. And this remains in the minds of people, and later on, it becomes an issue for decision making, considering the origin of the products and productive system. I believe, if you allow me as a last comment, at least in Mexico, I don't know how is this in Brazil. In Mexico, we still lack a lot of information regarding manufacturing and biosimilars in general. A lot of people do not know actually what a biosimilar is, type of what is biotechnology, how is this used. So this information has to be provided to population, not in a technical level, but the possibilities we have with the use of new technologies to produce a biosimilar of good quality and reduced cost. Oftentimes in Mexico, we try to filter the use of endemic drugs for endemic diseases, and the use of a biosimilar that the clinician do not know and do not have proper information. So we have to push a little bit, so we have a more openness in the use of biotechnology and biosimilars. So the end user receive more information, can investigate a little more, and understand through this information that is directly provided by the Department of Health and governments in general, and universities that have more opportunity to communicate from the scientific centers that biotechnology is safe and has many advantages. We still lack a lot in this area, at least in Mexico.
Any other statements? No, I agree. It's very similar here in Brazil, and Tiago probably agrees with me. We still need a lot of information. The fact that we're having a scientific discussion in society is very good because it gives people a perspective of where we are, but the opposite is also true. Fake news and misinformation that comes along with it. And a part of our role as a supplier of technology and as a manufacturer is also to inform people and society of these things. Definitely, and we have to remind people that before, we didn't have digital influencers who are now using a much simpler language to speak to society about products, therapies, and what people are doing. So there is a lot of debate in society to provide more information if you work in this area. So that was the last question we had in our conversation. It was great. It was great to talk about this with our audience. And I'm not sure if we have any time for questions from the floor. No, we are a little bit late, so we will have to continue. You did it very well. Tiago, Mariana, and Martin, thank you very much. Let's give them a hand. Thank you for this great discussion. Thank you for your comments, and we'll refer back to it during our summary. So we will continue with the next discussion, and I'd like to invite Manish, who is a Senior General Manager for Wockhardt in Brazil.
From Biocon also. He's based on India. It's 9 p.m. right now. So it's great pleasure, and thank you, Prasad, to join us. I know it's a little bit late for you. Let's see if we can connect it. He's going to be the moderator of that.
Yes, thank you, and good morning to everyone. It's a great pleasure to be part of it. I am trying to start the video, but it is, I think it's working now. Can you see me and hear me?
Yes.
Thank you so much, and pleasure to be part of the conference. And in fact, it is my first virtual conference in Latin America. So I want to thank you all for that. And I want to welcome Mr. Manish here. Mr. Manish and I go a long way back. We also had a pleasure of working together when Manish was a very young man. And when I returned back to India, Manish was spearheading our biosimilars business for the Latin America. Later, he took on many more big assignments. And Manish has grown into a full-fledged senior executive in the Latin America. And I would like to host this conversation for all of us to learn more from the experiences and all the initiatives that Manish has taken. And I hear one saying, cannot hear, I'm sorry. So I request the host here to say cannot hear. I hope everyone can hear me here in the conference.
Yes,
So,
yes, can hear you. Loud and
yeah,
clear.
so thank you, Manish, for joining. I think as the topic has been given to us, how about the biosimilars business, that too also in the tender business in Brazil? I also want to start this conversation for Manish saying he was spearheaded the entire Latin American Brazil business for Biocon, a global multinational, into Brazil. So with that, I welcome Manish. And why don't we start, Manish, with the first question. Tell us about your journey in Brazil and how is your pharma landscape, and give us a little bit of background of how the biosimilars is shaping up in the Brazil.
Yeah, thank you, sir. Thank you so much for such a nice word. And being my mentor, I'm really honored to be here. You are today moderator, sir. So as a journey, if you see in the Latin America for me, like I worked in various countries, right from the Guatemala, based in Guatemala, Lima, Santiago, and then Brazil with the Biocon, and recently with WorkHard for last 5.5 years. Very interesting market. I have worked a different market and found to be the Brazil being one of the largest market in pharmaceutical sector. If you compare global market, if you take here, compared with the USA, China, Japan, Brazil is one of the top 9th market in pharmaceutical business. Accounts to be around 25.5 billion market, and a very high rate. In the top 10 list, it is growing. China, which is growing by 9.5%, and Brazil, which is growing around 9%. The market, if you see, it is devalued in terms of US dollar because since the local currency has devalued here. Otherwise, the market, if you see here 2 or 3 years back, when $1 US was 3 reais, the market was around 33 billion US dollar. So you can see here the immense potential Brazil have in terms of pharmaceutical sectors, right from the finished product, biosimilars, and also in generic products. So anyone who want to have exploring a global market in you go right from going to the emerging market, taking the China, India, I strongly recommend for the Brazil market entry. I know Anvisa is very strict for the entry, but when you have authority is strict here, so entrance is also tough, but you get a premium price, a good market here. The few selected companies are there who can provide a right documentation, right product. So this is all I can share for the Brazil market, sir.
So Manish, that is a very good start, and you are giving a very good overview how the Brazil is growing the market. And in fact, you are saying is it is bigger than India and China. So that's a very good news for the pharma sector. Why don't you give us a little bit more about how the Brazilian pharma market is split between, as you said, the $20-30 billion? How about the retail business and the tender business? If you can give some insight about that.
Yeah. So if you see here, like social security coverage in Brazil is around 50 to 55%. So still there is a huge chunk of market in the retail segments. Brazil, if I compare with the emerging market like India, China, Brazil is still a market of prescription products. Branded generic, we say, branded generic. Local companies are very strong here. You take all the giant companies are there, EMS, Ashe, NIPS, all these companies are giant and they are very strong in the branded generic, are also providing a generic products. So retail segment is strong. Retail, if you see here, even the prescription is available in Drogaria Haya. They have one retail is like 2,000, 3,000 retails are there. The tender market, if you see here, it is coverage is 55 to 60%. Like if I compare with any other Latin countries like Costa Rica, Colombia, where the social security is very strong. The coverage is around 90 to 95% in that countries. Brazil still needs to make a better coverage to their population and social security. But yes, the market is immense potential for both retail segments and the tender segments, depend upon what kind of products you are coming in the market. If you are coming with a product which is related only to the tender market, like I say for my company, human insulin is there. It's the completely commodity products and it's a completely tender market for Brazil. Government floats around 20 million vials, you participate. And if you have capacity to supply, you participate in tender and supply 100% of that.
So that is also, I think, very good news for all the pharma companies if they want to go into the tender space as well, Manish, right? And
yes.
As the onset of biosimilars, you have led the first trastuzumab, as I know, into the biosimilars market. Why can you share some light on how the tender business works for the biosimilar? How the market operates? If you can give some idea about the biosimilars tender market.
If I tell you that biosimilars, especially if you see trastuzumab, the high value products are there. So the main buyer is the Ministry of Health. Human insulin, like if you see diabetes, it's one of the Ministry of Health, like Brazil being the fourth largest in diabetic patients. It's around 12 to 13 million diabetic patients are here. The first is China, India, USA, Brazil. What is the plan of the government is to have a proper coverage of diabetic patients. So annually the Ministry of Health, SUS, what we say, Unified Healthcare System, they float a tender. Talking about my product, I will tell you, it's a 20 million vial tender is there for human insulin. You participate in that tender, you quote a price, a very open tender is there in Brazil, like online tender is there. You participate, you quote a price. If you are a winner, you supply to the Ministry of Health.
So tell me, when the tender is won, is it, if there are multiple players in there, is it how many awardees are there? Is it given to the only one player, the entire tender, or it has been distributed by the government to multiple parties?
Yeah, that's a very interesting question, sir. And I have experienced here for that kind of things. The good part is like tender is floated. It's three part of tender is here. One is SUS, the federal tender is there, which have a huge quantities. The second comes the state tenders. All the state governments have their own budget to participate in tender. And then there is the municipal tender, the small tender in the municipal sectors, they have their own. So a state tender which is floated once in a year, You participate, it's an online tender, you participate in that tender, you quote a price, and the best price is a winner. Now you're coming with like multiple tender. I can participate any like 5 companies. Whose price is less, they're the winner. There is also option in the biosimilar, like they give you, suppose 20 million vial tender is there. In the 5 million, they will give you to this very small company that whose turnover is a 1 million. But I had suffered in that because I can't supply 20 million. So I was trying to supply the 5 million. But you participate through small companies. So there the budget is not there even to have us buy out the 5 million vials. So that is the where we find a little bit problem. So ultimately it goes to a bigger companies who can supply the whole quantities at the lower price. Like I can share one experience because I have also worked in Paraguay, a very small country. It is there in Paraguay. The good part of that country in the tendering system is that they float the tender, and there are 3 winners. The first winner is like who is having the lowest price. 60% of the volume will go to that, which is having lowest. Then the second lowest priced company will be given around 30%, and the third lowest price will be given 10% of that volume. So at least 3 companies are there. The government is also secured in the supply. We are also like as a company who participate. Okay, if I am not getting the whole quantity, I'm getting a chunk
60,
of
you can get a 60, 30 or 10.
Yes. So that is the one part where I recommend for SUS at least to create that kind of tendering system in Brazil, where at least 2-3 companies can participate and provide a complete volume, and coverage will be better as compared to giving it to one company.
So Manish, you bring a very interesting point here. What you are saying is Brazil has the 3-tier tendering system, whether it is a federal tender, the state tender, and municipal tenders. While there are the 3 tender systems are there, but winner takes it all. However, like in another countries such as Paraguay, if there are 3 awardees, there are different volumes there, thereby securing the supplies in case the first tender winner does not take it. So it's a very interesting combination across the Latin America landscape as how each country operates and how they take care of their nationals and the health security, I would call it. So I will under the health security as well. Go ahead. Yes, you wanted to add.
I will add here, because due to that process, I had suffered here in the tender because our biosimilar, it was registered 10 years back when the biosimilar regulation was not there. So what happened? As a participant, I participated in the big tender, but my product used not on pediatric patients, only on the uses of adult. But while the multinational company, they were having used on the pediatric adult.
Dual therapies, pediatric as well as adult.
Yes. My price was lower than the other company, but somehow I was disqualified from the tender because it was in the use of pediatric patient. So what is my recommendation here? Okay, you don't take 20 million from me. You take it maybe 15 million or 10 million from me. For the pediatric usage, you take it. How many pediatric patients have in diabetics? 1%, 2%.
Yeah,
so you are losing that. That kind of consideration has to be taken in Brazil.
So that you bring a very important point. What you are trying to say is the qualification in this case was the pediatric indication as well as the adult indication. And although you have given the very competitive pricing for the tender, you lost out because although only that 1% of indication was not met in your dossier, you lost the entire 99% of tender as well. Right. See, that brings a very important and a very interesting next question from my side is, why don't you educate us, is what are the qualification process for the tendering for any company to participate? Do you need to have a manufacturing facility in Brazil, or can a foreign company does a foreign direct investment? How does the qualification work, either from finances or from the dossier perspective?
Yeah, in the tender process, like if I compare, take two countries, Brazil and Mexico. So what Mexico have, Mexico, if you are getting from local manufacturing, so your tender participation like increases like 60%. Like Brazil is open for any companies, any countries. If you have registration of the product and you have local entity, so you can participate in that tender. It's not that local will be given a preference, you are locally manufacturing. No. Equal preference has been given to any countries, whether it is coming from India, China, Europe, USA. But you need to have a local entity here. If we take a small molecule product, the only thing why the local entity, because you have to go for a quality control release of the product here. This is the one regulation in Brazil, like mandatory to have quality control release. Unlike biosimilar quality control is not released, but the way Anvisa is very strict in the terms of the temperature, 2 to 8 degree, you have to maintain the temperature. Data logger has to be there. Then that data logger temperature has to be sent to Anvisa to release the product. So, sir, can you repeat the next?
So what I wanted to ask is, one is you talked about the data logger, you talked about the dossier requirement. See, the one good important point you mentioned is, unlike the Mexico, here you don't have to have a manufacturing facility. The registration has to be there. So that is a very valid point. But how does this work then when you do the PDP, where you have to transfer the technology and manufacturing in Brazil?
Right, sir. If you say here short-term tender is like a 1-year tender, which is given by the SUS, federal government. Good part about Brazil, they have come over with the product development partnership, like especially for the complex
which is also called as PDP. Yes,
PDP. So you need to have a local manufacturing here, but the government has what they have given, like down the line, they will give you a certain percentage, like suppose the human insulin 20 million vial. So you get a 10 million vial for the PDP, but in that period, like from year 1 to year 10, you have to make a phase-wise technology transfer in the Brazil. It is good for the Brazil. Technology are coming here, locally manufactured here, from finished. Slowly it will go to finished, then the naked vial supply will be there, and down the line, after you make in 5-year transfer of technology for finished, you go for the API. Another 5-year, the API development, API technology transfer, and in 10 years time, the local manufacturing will be there. So it is a long-term business. I'm also transferring the technology to the local companies and to the local government laboratory here. Basically, we have to combine with the government laboratory like Biomingens, Pharma-Guinness, Funet is there. So we have to transfer the technology to the government laboratory, as well as you have a partner here as a private partner, you can associate with them also.
So it is like a public-private partnership development program. What also you made a good point is, while the SUS tenders, the federal tenders are for the short duration, which is a 1 year or 2 year, depending on the tendering process. Whereas this partnership, private government partnership programs can go all the way to the 10 years, and you have an assured business for 10 years. It's kind of a win-win strategy. What I understand from you, Manish, is the Brazil as a country gets the technology, in-house manufacturing, creation of the economies, whereas the pharma companies get an inroads and sustainable business into the country. Is that what the whole program is all about?
Exactly. So, sir, I will give you one example also with my colleague here sitting from Biocon. Trastuzumab, when it was there, like the price at that time from the multinational is worth $3,000 US. And then the Biocon came here when the price with the PDP, the price definitely it's like a premium price, but from 3,000 you are coming to 1,000 US dollar, and you are getting a sustained business for 10 years. Transfer the technology, and there is a group here, like if you say Bionovis, Horizon, they are working, 4 or 5 companies are getting together, and they are working with all the, like Pfizer, they are working with the Pfizer, Janssen and Janssen, to get the product, complex molecule here, transfer the technology, a shared business I am giving you, and then locally manufacture here. In this way, like government is getting a manufacturing here for the complex molecule, you are reducing your exchequer in the government budget. Like tomorrow you are paying to $3,000. Today you manufacture locally and you buy it from the local market, you pay $500. So you are reducing the cost from the Ministry of Health.
So you are saying that is the cost of healthcare spending on the government significantly reduces on that, right. And there is a consortium of all the companies who are working with the bigger. Now, one question I had for you, Manish, here is, now you mentioned, you gave like that insulin example of 20 million, but if you do the PDP, the 10 million is carved out. So are you saying if the PDP happens on certain complex molecule, that much volume is kind of reserved for those partnerships, so that is reduced from the total tendering process?
So that is a very tricky part here. Like, yeah, the government tells that 10 million annually to buy, but it depends upon the Ministry of Health. Like they can buy 10 million or they can buy 8 million. So what, as a company that I have to also secure myself. So what we do, normally we put it on minimum quantities to be purchased by the government. Like 10 million, okay, you buy not 10 million, you buy 8 million. Below 8 million, if you buy, then okay, I will think of transferring my technology to you. So that is the one condition as a company, because I have to also protect myself. So we put a certain condition also. Yes, there are ordinances here, like they can, like Ministry of Health, they can surpass that also. But as a company, that also I have to secure myself. Okay, I'm transferring this technology. Down the line, it will go this technology, but I have to earn as a corporate. And a company, we are sitting here to earn also. So
Manish, very, very good point. What you are trying to say is, if the partnership has to be the mutually beneficial for the company, the company with the technology, the recipient company, and the government. So there is a less money, as you said, the less on the exchequer of the Ministry of Health. And when this three comes into a symbiosis, that's when the real partnership happens. Otherwise, one loses out into the whole process. Just a time.
Yeah, go ahead. Sir, you can also, in this way, important part, or like you are getting a very high cost molecule, a complex molecule transferred here down the line. We as a corporate, we are also secured, and as a Brazil government, you are also getting a new technology where if one product technology is there, definitely Brazil have good R&D, good brains here. They can develop other molecule from that. So Brazil, what require, like in the innovation sectors, if you want to be in a top like a China, which is growing very fast, because they are good into innovation and copying. So you have to do it, like you have to transfer the technology here to get at the local innovation.
So that's very good point. Manish, since you are physically there and I'm online, I just wanted to do a quick time check. I don't know how much time we have been allotted. I would love to have this conversation, but I want to, reserve some time for our audience as well to ask the question. But before we do that, I want to know, Manish, any closing comments you have, because it's an exciting journey. You have participated, you shared some success stories. You also shared your part of the, not so successful stories and challenges. So any closing comments before we open it for the audience?
What I can say is, especially in the complex molecule, Brazil requires this product. So Anvisa is very strict. Anvisa little bit has to be lenient in this kind of complex molecule. Like one example I am giving, a Glargine, a Lantus brand. we tried to register here, but somehow it was rejected by the Anvisa because there was one in the study, our clinical trials outlier was there. So, but that study was done in the USA. So this kind of things Anvisa needs to take into consideration. Little bit Anvisa has to be lenient in giving a registration. I'm not telling any company can come and register here. It's good that Anvisa is very strict. But yes, for the complex molecule, for any kind of complex molecule, pegylated filgrastim, glargine and all that, little bit lenient in the documentation part. It is benefit for the government only because you are paying like a glargine. It's a market of 120 million US dollar. And the government buys around 20 to 30 million US dollar. And if some more of competition is here, you are reducing the price for the benefit of the patient and the population.
That's fantastic. And as you have explained it in a very well manner, but I still think there are a lot of complexities and a lot of experiences required to operate into that market. So thank you for, sharing at least some light on it and making it little easier. So while we now turn on to the questions, I have online one question which has been asked is, what is the process for inclusion of biosimilars in the tender? So that's one question on online at least. And then we'll also go to the actual in-presence audience there as well.
I think there is no such process. Like if you have registered here and there is a demand in the Ministry of Health, you participate in that tender. Like you need to have local entity and you participate. It's online, very open. It's nice about Brazil. It's an open tender online. I quote $100.
You have to have a registration of the product, and you need to have a local office in Brazil, and it's an open tender after that.
Right. Yes. If it is there in the government list, they float the tender, and you participate in that tender.
Excellent. So I turn on to the audience. Any questions from audience, please? It's been a great conversation.
Thank you, sir. Thank you.
Thank you for that. Let's see if you have any question from the audience. No? Okay, thank you so much for that.
Thank you so much.
That's great. That's great discussion. I think that we just anticipated or just put some curiosity here about the PDP that's going to be discussed tomorrow morning in a session with Tiago, and also we have someone from Samsung. We have a good discussion prepared for tomorrow about the PDP, a success story in Brazil. It's going to be great. You anticipated this kind of factors that is very important here in Brazil, the PDP. So thank you so much. Thank you also for Prasanth to stay until late night with us. I know that it's late in India. Thank you so much to join us.
Thank you.
It was a great pleasure. Thank you, sir. Okay. Bye. Thank you. Bye.
Bom, agora a gente vai para.
So now we will have our last session of this morning. It is also very important. We will hear from Ana Carolina from Leaps, the extrapolation of indications with biosimilars. Please, Ana Carolina, you have the floor. Ana Carolina Cardoso.
Bom dia, tudo bem? Todos me escutam?
Good morning. Can you all hear me? So, good morning. It's a pleasure to be here with you. I know that this is the last presentation preventing you to go to lunch, so I'll try to be brief in the presentation. If you have any doubt, feel free. I will be here during the afternoon. You can talk to me during the afternoon as well. So, the topic of my lecture today is biosimilars and extrapolation of indications. I'm Ana Carolina Cardoso. I represent Leaps Pharmaceutica. I coordinate the MSL team of Leaps. Here, my disclosure. Excellent. Well, this first slide is more to think on the time we already have both of regulatory practice as the use of biosimilars. If we see an overview of the guidelines in the world, in 2005 we had the first guideline approved in Brazil in 2010. So we can think that we have almost 20 years of experience in Europe, in Brazil a little bit more than 10 years of experience. And what I want to ask you, have we really exhausted all the knowledge necessary for biosimilars? Well, in Brazil, to give you an idea, thank you, I believe it is not working. To give you an idea of a process for approval, as I said, in 2010, we have the first legal framework defined in Brazil, and as of 2015, we have the two first biosimilars approved to be commercialized in Brazil. So focusing in oncology and hematology, that is where LIBS acts, we have 15 biosimilars. And in 2017, we have the approval of the first oncological biosimilar in Brazil in partnership with Bayern. That was approved in 2017, and in 2019, the first with 100% national production. In the next slide, I try to discuss the knowledge about biosimilars. This is a study we developed in the medical area, in the healthcare sector, together with a researcher from Rio de Janeiro, to understand the perception of Brazilian oncologists in terms of the practices and concerns of using biosimilars, especially the trastuzumab. That is the one we had approved at that time in 2019. And in these two charts to the left, we have the perception of use. So 96% of the clinicians answering the survey said they feel comfortable in prescribing trastuzumab biosimilar, which is very similar to the survey conducted in Europe, also in 2019 by EMA, where doctors, generally speaking, here is not only oncologists, but about 86% feel comfortable in prescribing biosimilars. So it's very high. Apparently, there is no concern on the use of biosimilars. But when we go a little bit more into depth, which is the chart to the right, when we talk about extrapolation, this percentage drops. When we ask if they are comfortable to use biosimilar to all the indications where they have reference drug, the level of comfort decreases. In Brazil, we had 63% of oncologists are comfortable in extrapolating, compared to EMA, where 76.7% feel comfortable to extrapolate the indications of biosimilar. In the next study, more recent one carried out in Belgium, has the same idea. Not only clinicians, but also pharmaceutical professionals, when question if they feel insufficiently trained for biosimilars, the percentage for biosimilars is much higher. So they feel they have less training for biosimilars compared to other molecules in general, which makes more evident that the field, industry should start initiatives, because although we have a long time of experience, both regulatory and practice, we still have to work with some specific concepts for biosimilars. And this is what I will focus on in my lecture. Then I go to extrapolation.
Em relação à parte conceitual dos biosimilares, eu acho que tem, obviamente, que todos aqui conhecem muito de biosimilares, a ideia não é chover uma olhada aqui para vocês, né? Mas é trazer também um pouco da minha perspectiva, né? E da perspectiva aqui que eu trabalho com o time de MSLs ao longo desses anos, trabalhando com os oncologistas e hematologistas principalmente. Eu acho que dois pontos principais em relação ao conceito de biosimilares que a gente pode sempre aprofundar tanto a parte de variabilidade, a variabilidade intrínseca dos produtos biológicos de maneira geral, e também a parte da totalidade de evidências que traz a parte conceitual dos biosimilares. Então, quando a gente pensa na variabilidade, aqui a gente tem um cenário visual comparando um produto de referência antes da aprovação, pós-aprovação e um biosimilar. 3 anticorpos monoclonais, que eles podem ter diferenças entre eles, porque isso é inerente da própria molécula biológica, né? E já ressaltando e reforçando aqui, quando a gente fala dessa variabilidade, a gente tem que ter essa segurança de que nenhuma dessas diferenças ela vai afetar o final do processo, que é a segurança e efetividade daquela molécula. Esse é um estudo bem conhecido de Weser de 2016, que ele mapeou, né, todas essas variabilidades intrínsecas das moléculas. Aqui a gente está falando de medicamentos de referência. Então, todas essas variabilidades, elas foram mapeadas de acordo com a porcentagem de risco. Então, a gente tem um baixo risco, como, por exemplo, uma troca de um filtro ou de meio de cultivo, ou do próprio fluxo laminar, que conferiria uma mudança que consideraria um baixo risco, ou um alto risco, como, por exemplo, uma troca de uma linhagem celular ou do próprio meio de cultivo. Então, baseado nessas trocas que podem acontecer no processo produtivo, todas essas variabilidades, todas essas variabilidades, elas são mapeadas. Aqui eu reforcei 3 moléculas, rituximab, o infliximab e o trastuzumab, para vocês verem como isso é variável, né? Como a distribuição dessas variabilidades e do risco também acontece entre as moléculas. Então, moléculas que podem ter variações de maior risco ou de menor risco, mas que isso, no final do processo, é controlado tanto pela indústria quanto pelas agências regulatórias. Então, a gente sabe que essa variabilidade inerente, ela acontece mesmo dentro de lotes do mesmo medicamento, do medicamento de referência, tanto do referência quanto do biosimilar, mas desde que tudo isso seja inteiramente controlado pelo controle de qualidade, pelas agências e pela própria indústria, para que não afete, não ocorra uma perda de eficácia e segurança no final do processo. Então, aqui no Brasil, né, como eu comentei, a gente tem a regulamentação através da RDC 55 da Anvisa, data de 2010, em que a gente tem a aprovação dos biosimilares, o desenvolvimento, né, dos biosimilares aprovados pelo nosso exercício de comparabilidade com relação ao medicamento de referência. Então, através desse exercício de comparabilidade, o objetivo é que o biosimilar ele seja aprovado de acordo com os mesmos padrões, né, de qualidade, segurança e eficácia que são aprovados e aplicados para todos os outros biológicos, né? A gente está de acordo com as agências, as outras agências, as principais agências regulatórias do mundo, a FDA e EMA, em que garantir, né, o objetivo é garantir que nenhuma diferença, nenhuma potencial diferença ocorra entre o biosimilar e a referência em termos de segurança, pureza e potência do produto. Então, de maneira geral, essa é a tríade que sempre a agência regulatória vai desenvolver e vai garantir, né, que tenha a mesma qualidade, eficácia e segurança. Esses são os principais pilares que as agências regulatórias trazem, né, nessa avaliação dos biosimilares. Aqui nessa aula a gente vai focar nos 2 primeiros pilares, que são os estudos comparativos ou a totalidade de evidências, que eu falei para vocês, a parte de extrapolação. Então, entrando nessa parte... Próximo, isso. Antes de entrar nessa parte da totalidade de evidências e dos estudos clínicos, um ponto também que é importante pontuar, com base também em tudo que é descrito na parte regulatória, a gente não pode confundir biosimilares com o que a gente chama de intenções de cópia, né? Então, são produtos diferentes, os biosimilares, eles passam por uma regulamentação muito mais rígida. Então, aqui, só de maneira comparativa, para a gente ter uma aprovação de um biosimilar, a gente tem que ter toda a caracterização comparativa, né, clínica e não clínica, os estudos clínicos, toda a evidência de segurança e qualidade e eficácia, e seguir também todos os requerimentos da Organização Mundial da Saúde e das principais agências regulatórias. E o que a gente muitas vezes não vê nesses outros produtos, que são os produtos chamados de intenção de cópia, que eles podem ter parte dessas informações incompletas ou insuficientes. Bom, qual que é o caminho que um biosimilar ele precisa ter para garantir ou para receber essa aprovação regulatória? Então, a gente sabe que é um caminho árduo, principalmente ali no início, que é justamente essa parte de engenharia reversa, né? Então, aqui a gente tem representantes da parte produtiva, né, do desenvolvimento de um biosimilar. Então, essa primeira início em que você tem que caracterizar diversos lotes do medicamento de referência até chegar numa molécula possível, né, de ser um similar, digamos que aqui é onde se gasta a maior parte do tempo e esforço mental, inclusive, para chegar nessa parte. Depois tem toda a parte, segundo, terceiro e quarto ponto, né, que é a definição das condições de cultura, todos os testes de função, entrar e conseguir encontrar a melhor formulação que vai ser desenvolvida para essa molécula, toda a parte de confirmação clínica, e tudo isso, todo esse caminho é que vai ser entregue para a agência regulatória, que ela pode, ali, nesse momento, decidir pela aprovação ou não aprovação do medicamento biosimilar. Então, no próximo slide, eu vou trazer agora um pouco mais exemplificado o que são esses passos, né, o que inclui todo esse conteúdo que é entregue para as agências regulatórias. Então, aqui a gente tem, principalmente, a parte das definições analíticas, que envolve tanto as avaliações de estrutura e de função. Então, por exemplo, a gente tem que garantir que esse biosimilar, né, garantir a comparabilidade, a similaridade do biosimilar com relação à referência em todos esses pontos, por exemplo, mapeamento peptídico, a sequência primária que deve ser 100% idêntica, todas as impurezas, mapa de glicosilação, que é extremamente importante, porque a gente vai avaliar também de efeito clínico depois, cadeia pesada das imunoglobulinas, todos esses pontos são comparados do ponto de vista estrutural. E do ponto de vista funcional, a gente tem também outros ensaios em que o biosimilar precisa passar em relação à referência, pronto, que são, por exemplo, as funções efetoras, função de ligação de receptor, a função de sinalização e tradução de sinal, citotoxicidade. Então, tudo isso é o que garante o que a gente chama de fingerprint da molécula, né? Um biosimilar, ele tem que ter o mesmo fingerprint do que já é conhecido por medicamento de referência. E aí a gente chega nessa figura que já é bem conhecida por todos, acho que essa é uma figura clássica quando a gente fala de desenvolvimento de biosimilares, principalmente na comparação também com referência. Então, acho que aqui a gente pensa também como uma quebra de paradigma, porque a gente está acostumado a pensar na rota de desenvolvimento de um produto inovador, como é representado pela figura da esquerda do produto de referência, em que você... os produtores nas indústrias, elas iniciam com essa caracterização analítica, mas à medida que os próximos passos os passos subsequentes vão sendo atingidos, o volume de estudos e o volume de informação, ela vai crescendo, até chegar, por exemplo, na parte dos estudos clínicos comparativos, em que é necessário se fazer todos os estudos para todas as indicações que se necessita, ou que se quer para aquela determinada molécula. E a mudança de paradigma do biosimilar é porque exatamente o inverso, né, que por isso é representado pelas pirâmides invertidas. Então, do biosimilar, a gente parte do princípio que a gente já conhece tudo que a gente já tem de caracterização daquela molécula de referência. Então, a gente não está mais olhando para uma necessidade de demonstrar uma segurança e efetividade per se, mas a gente precisa demonstrar a similaridade com o produto de referência. Então, por isso que a base aqui da pirâmide, que é a base analítica, né, toda a parte de acesso de estrutura e testes funcionais, como eu mostrei para vocês, é onde a gente vai demandar a maior parte do tempo de esforços até chegar no topo dessa pirâmide, né, passando pelos estudos não clínicos, pelos estudos farmacológicos, de farmacocinética e farmacodinâmica, até chegar no topo da pirâmide, que é um estudo clínico numa indicação sensível, né. E próximo isso. A indicação sensível, então, eu trouxe esse slide mais para reforçar essa mensagem do que é a indicação sensível, né? Ela pode ser qualquer uma em que é possível verificar qualquer potencial diferença em relação à função do biosimilar comparado com referência. Então, por exemplo, as barras da esquerda, a gente mostra uma população que ela é pouco sensível, porque o que eu tenho de resposta na barra verde, o que eu tenho de resposta em utilizar o meu produto inovador, por exemplo, é muito baixo, né? O benefício que eu tenho ali é muito baixo com relação ao tratamento padrão ou ao placebo. Então, essa seria uma população pouco sensível, porque se eu comparasse nessa população biosimilar com um medicamento inovador, qualquer potencial diferença seria difícil de visualizar, já que eu não tenho nem com o inovador um benefício fácil de ser comparável com relação ao tratamento padrão. Já na parte direita, quando a gente vê as barras mostrando uma população, no slide anterior, por favor, quando a gente tem uma população mais sensitiva, a gente vê que essa diferença em relação ao tratamento X, o inovador, ele já promove um benefício muito maior em relação ao tratamento padrão. Então, essa é sim uma indicação potencialmente sensível para prever qualquer diferença entre o biosimilar e a referência. Então, os estudos clínicos, né, por que a indicação sensível é importante? Porque a gente evita com que esses estudos clínicos eles sejam repetidos desnecessariamente, né? Então, uma vez que a gente tem uma indicação que a gente sabe que ela é sensível, a gente pode evitar essa repetição de estudos. Então, desde que essa indicação sensível eu consiga mostrar essa comparação de referência com o biosimilar, eu consigo compor essa pirâmide, né, da totalidade de evidências, somando aos meus estudos não clínicos, toda a base de comparabilidade analítica, e dos estudos clínicos. Então, o que a gente tem aqui é o que a gente fala que é uma velha droga feita por um novo processo de produção, né? Então, a gente já conhece tudo o que veio, que tem de literatura do medicamento de referência, agora a gente só precisa mostrar que o biosimilar, ele tem o mesmo perfil de qualidade, segurança e efetividade. E nesse processo de produção, esse ponto é importante também, que não a dificuldade, mas qual que é a robustez, né, no desenvolvimento dessas moléculas, dos biosimilares, ela é dada passo a passo. Então, a cada passo daquela pirâmide, né, que a gente estava mostrando, eu preciso garantir que todo o rigor científico utilizado naquele passo, ele vai assegurar o caminho para o passo subsequente. Então, a gente vê com isso que a redução dessa incerteza com relação à similaridade, ela vai decaindo ao longo desse processo de desenvolvimento dos biosimilares até a gente chegar nos estudos clínicos, né? Então, a gente tem que garantir que, por exemplo, os estudos clínicos, eles tenham sensitividade e especificidade o suficiente também para conduzir essa comparabilidade, olhando para o design, para o desenho estatístico, para a população escolhida, como eu falei, da população sensível, e também para a escolha dos endpoints, dos desfechos. Então, essa mudança de paradigma, a gente fala, não no sentido de que eu não devo olhar para o paciente, mas ele troca esse olhar do paciente para a droga, né? Quando a gente olha para um medicamento inovador, a gente quer demonstrar um benefício daquele novo medicamento para aquela indicação, e a gente quer demonstrar que os desfechos clínicos vão ser melhores com o medicamento inovador do que o que eu tenho para o tratamento padrão. Quando eu olho para um biosimilar, e é aqui que está essa troca do paradigma, a gente tem que olhar com um olhar científico de similaridade, né? Eu não espero que eu vou ver um benefício ou um desfecho melhor com o biosimilar, eu espero que eu vou ver um desfecho tão bom quanto o que eu já conheço para o medicamento de referência. E essa tabela, eu acho que ela reflete também de maneira comparativa essa mudança de paradigma quando a gente olha para o processo de desenvolvimento de referência e de biosimilares. Então, para o medicamento de referência, a população escolhida ela pode ser qualquer uma que se queira provar, né, o benefício do tratamento com a molécula inovadora. Já para o biosimilar, a gente tem que olhar para uma população sensível, uma indicação sensível e mais homogênea para garantir que qualquer diferença seja captada entre a comparação biosimilar com a referência. O desenho do estudo também, o medicamento referência, a maioria das vezes são estudos de superioridade, então eu vou trazer a comparação da minha intervenção com o meu standard of care. Para o biosimilar, a gente fala de estudos de equivalência ou não inferioridade, em que o meu objetivo é avaliar head-to-head a comparação do biosimilar versus o medicamento de referência. Quando a gente fala dos outcomes, dos desfechos, dos dois, né, tanto para o biosimilar e referência, a gente tem que olhar para desfechos sensíveis e desfechos de eficácia e segurança. A diferença é que, para referência, muitas vezes o foco são mais em desfechos long-term outcomes, né, desfechos de longo prazo. Diferente dos estudos de biosimilar, que a gente olha para desfechos curtos, como, por exemplo, taxa de resposta objetiva, farmacocinética, farmacodinâmica. Em relação à segurança e imunogenicidade, os dois também fazem avaliação de risco e benefício, com a diferença de que, com referência, a comparação vai ser sempre com o meu standard que tem para indicar. Eu tenho que comparar esse risco e benefício, segurança e a imunogenicidade com relação ao que eu já conheço para o medicamento de referência. E aí, entrando na extrapolação, que a gente traz que não é possível para o medicamento de referência, mas é possível sim para os biosimilares, baseado em evidências científicas. Então, a ideia é de que quando a comparabilidade de um biosimilar ela é demonstrada em uma indicação, então a extrapolação dos dados clínicos ela é possível, né, e ela é aceitável, desde que a gente siga as principais justificativas científicas, que são essas aqui à esquerda, mostradas no slide. Então, a gente tem que garantir a similaridade com relação à análise físico-química, análise estrutural, todos os testes funcionais in vitro e in vivo, todos os dados que a gente tem de farmacocinética e farmacodinâmica, e os dados, claro, de eficácia e segurança. Então, a partir do momento que eu tenho essa totalidade de evidências, os dados clínicos numa indicação sensível, eu consigo extrapolar para as outras indicações que eu já conheço a partir do medicamento de referência, que elas funcionam tão bem quanto. Com os pontos de consideração, então, claro, as agências, eu trago até no slide os símbolos, o que é referenciado também pelas agências regulatórias, então a extrapolação é baseada nessas justificativas científicas que eu falei para vocês, com alguns pontos de consideração, então olhando sempre também para a sensitividade do estudo, né, de novo, a indicação, os desfechos escolhidos, para o mecanismo de ação, então o mecanismo de ação ele deve ser o mesmo, né, do referência com o biosimilar, para que a extrapolação possa acontecer, e alguns requisitos específicos, como, por exemplo, a imunogenicidade, a farmacocinética, posologia, que pode mudar de uma indicação para outra. Exemplo, uma indicação autoimune para uma indicação oncológica. Então, tudo isso é levado em consideração pelas agências regulatórias na hora de aprovar a extrapolação do biosimilar. E aí, qual que é o motivo, né? O porquê extrapolar? Extrapolar é importante, né, e o principal objetivo da extrapolação, ela se dá para a gente tentar evitar a repetição de estudos clínicos, a repetição desnecessária de novos estudos clínicos, baseado no que eu comentei para vocês, que a gente já conhece, né, toda a base científica da molécula a gente já conhece. Então, a ideia é que a gente não sobrecarregue o processo de desenvolvimento e que a gente também consiga reduzir tempo e custo para chegar com uma nova opção, tanto para os médicos quanto para os pacientes. Aqui é uma tabela comparativa também dos principais pontos que eu falei, então comparando, a Anvisa ela está muito de acordo, né, com que as outras agências regulatórias também, aqui eu trazendo comparação à MFA, então todas elas pedem, né, para uma aprovação de um biosimilar, tem a exigência de todos os estudos comparativos, a extrapolação ela é aceita, tem a exigência também da farmacovigilância e a intercambiabilidade, que é a única que difere com relação à FDA, que eles trazem como possível, porque lá eles têm, né, uma regulamentação específica até para as indústrias que querem fazer um desenho específico de intercambiabilidade para esses biosimilares, e a Anvisa segue o que a EMA traz também, que é a escolha do médico. Então, o médico é que detém a decisão de fazer ou não essas trocas, tanto troca do biosimilar por referência ou troca entre biosimilares. E aí, já chegando no final da nossa apresentação, eu concluo como eu trouxe no início para vocês a pergunta, né? Por que será que a gente ainda tem, será que a gente ainda tem área, será que a gente ainda tem espaço para falar sobre biosimilares, conceitos de biosimilares? Por que será que ainda existe tanta preocupação? Uma das coisas que a gente discute bastante no nosso time, que eu acho que essa imagem representa bem, é o olhar que a gente tem para os biosimilares apenas para essa parte de avaliação clínica. Né? Então, por isso a comparação com o iceberg, a gente está olhando a ponta do iceberg e às vezes esquece que a maior parte do desenvolvimento ele está para baixo, né? Está para baixo da água, que é a base da pirâmide. Então, o biosimilar ele tem um esforço de desenvolvimento e a parte do racional científico, ele é muito grande também na base. Então, um biosimilar ele não é aprovado só quando ele chega no estudo clínico. Como eu falei para vocês, a gente tem passo a passo essas aprovações e esses requisitos de avaliação que culminam no exercício de avaliação clínica. Só mais um, por favor. Então, a totalidade de evidências é garantir para a gente.
So the totality of evidence leads to a more reliance in biosimilars, but we have to guarantee that quality, efficacy, and safety not only based in studies, but in the totality of evidence. And last slide, my take-home messages is a summary of what I brought. Biosimilars are highly of high quality and have the same profiles as any other reference drug, any other biological drug. The comparability studies include both analytical, non-clinical, and clinical studies to demonstrate that the efficacy, safety, quality are similar to the reference drug. Biosimilars contribute to sustainability of the healthcare system. So with them, we have more options to treat the same for the same indication. Extrapolation avoids unnecessary repeat of clinical studies. As long as we are able, with that, we optimize the time to produce. And the whole approval process has to be based on science, not emotions. We have to base in the quality of evidence. The scientific rationale is very clear to develop biosimilars. So we have to take this information and talk to healthcare professionals to have this greater level of comfort to prescribe biosimilars. Well, this is my contact, my email. If you want to send anything or ask, as I said, I will be here if you want to talk to me after. And I thank the opportunity of this invitation and have a nice afternoon. Thank you for your presentation, Ana Carolina. Well, if we don't have questions in the audience, I will give you one. Do you have figures like in gross figures of how much professionals discuss what is a biosimilar, what's their appetite to discuss and to understand the scientific basis to develop a biosimilar? Well, in this study, we interviewed 144 clinicians. And the EMA has a panel of 250 or 300. First was more for breast cancer, for trastuzumab, and now GI. But in a daily interaction with the clinicians, when we ask them about the perception, they say, "Oh, I have no problem with biosimilar." But when we start to bring the details, especially variability, these are points that are not clear for them, are not in top of mind. I believe that 30% still have that doubt, that they know how to ask, actually. We see this clearly in our lectures, in our talks, and I feel that some specific points for paradigm shift we should work on, and we still have room for that, for clarification. We have a question here. I'm Daniel from Sandoz. Anna, congratulations for your presentation, very complete. Well, you probably have followed the UK regulating authority that changed their process of approval for biosimilars, decreasing a lot the requirements for clinical studies. This was based on the issue that most of the molecules they evaluated there, 95% of them had any information added in terms of immunogenicity in the clinical trial based on compared to what it was studied in the preclinical trial. And in the 5% molecules that where they had the impact were processes from before 2010. My question is, in your opinion, do you think we will reach a point where there won't be need to include phase 3 trials, but not PK/PD? The end. My question, what is your opinion if will there be a moment when we no longer need phase 3 confirmatory trials? Actually, what happened in the UK is an example of what can happen in other places. I still do not see that in the short run to not require clinical trials in the evidence package to be presented in the dossier, but this reinforces what I mentioned about the totality of evidence. So more considering analytic comparability and PK/PD trials than the clinical trial that we know will take longer. We have an increase in cost because a clinical trial is very expensive. So the trend is the midterm, in my opinion. Work a little bit more this concept that to approve a biosimilar, I don't have to check just the top of the pyramid in a phase 3 clinical trial, but to the basis, to the comparative studies. And with that, but we have to work more in this concept for biosimilars. We have another question.
You have your answer. Like I've heard, if your PK/PD study is strong, so you don't require to do immunogenicity. It's a regulation now. I've heard that if you have a strong PK/PD study, and you don't require to do immunogenicity. It's difficult, but the company can manage a strong PK/PD study. So Anvisa accept. I may be wrong. I'm not a technical person, but I've heard.
Sua pergunta se a Anvisa requer
Your question is if Anvisa require the robust PK/PD study and if you need to carry out immunogenicity. Actually, for Anvisa, we do need not only PK/PD, but the clinical trial with evaluating immunogenicity compared to the reference drug. Well, thank you, everyone. It was a little bit longer session this morning. Ana, thank you for your presentation. Well, now we will have 1 hour break for lunch and seize this opportunity of being together during lunch. We will resume at 2:40. Okay?
Se eu não tiver mais maionese, sabe? AWS, é legal. Mas é tipo, você falou assim, como coordenação, Não, verdade. Se tá saindo aqui, era pra... Exatamente. O dele mesmo, eu tá mandando. Agora eu vou jogar pra imagem dele. Ué, qual dele fez? Eu acho que ele habilitou a saída do painel, só habilitar a saída do monitor. Não, talvez se faça para mexer alguma coisa lá. É que ele ficou da inspetor. Ele foi igual a você outra vez. Qual que é a resolução da saída do papo?
Ok, explica para essa história. Só pergunta qual é a resolução que ele está usando. Para você colocar para poder passar para ele aqui. Ela tem 60 Hz, mais 60 Hz.
Vamos mudar.
Yes, I think they get through this. Mas então, eu acho que eles não. Mas bem, a vez que estava aqui, ele está por aqui. Não, uma coisa é isso e a outra
É exatamente isso.
No, it's fine. I can show you.
Se ele disputar um HDMI, tá vindo para mim, jogar de novo, volta.
This on the cable. Esse é o único. Não.
Então, antes de eu fazer esse reguladores para cá, a primeira experiência, e você. Um Guarulhos do Rio, que é uma das experiências de prática. That's not very, we learn a lot. Nós precisamos de 1 minuto a mais, só para poder estar aqui. Então, ainda tem uma última chance de vocês darem alguma dica para quem de fora.
Hello?
Alguns lugares,
Hello?
Sorry, you didn't explain very well. I'm learning for this.
Okay. For each, maybe black bean and more meat and maybe creations. You can try so better day and day after. So not today. Yeah, the day after. Well, the 15 because that was yeah.
And so Eduardo and the cookies, it's good one. Yeah, and some kind of some like because you need them back after that.
Hello?
Yeah.
Okay, but I do have a deal.
Close to here, there's one thing that's muito bem, para quem é de fora aqui de São Paulo, hoje agora tem um show ali. Se alguém quiser assistir, acho que não tem mais votar. Não, o show São Paulo vai jogar contra o Flamengo. E tem uma ópera lá no mês do Corinthians, e acho que vai ser mais difícil. Flamengo joga o São Paulo. Pode ser um show. Nós temos um problema técnico na conexão aqui, a gente vai precisar de 5 minutos mais, porque vai ter que reiniciar o computador. Eu escolho para vocês pelo problema técnico, 3, 4 pessoas têm que ter arrumar alguém. Então, acho que a gente dá 3, 5 minutos mais e depois volta.
Hello?
Só que ele vai chegar no meio da manhã, acabou adaptando um semi-integral. Pedagógico, no ano dele só tinha tarde, não conseguiu domingo. Aí, às 8, cerca do mundo do Estado, que é o primeiro domingo, e aí ele estava perdendo a parte pedagógica à tarde. Ele estava morto, de cansado, dormia no meio da aula. Ele estava pagando isso para as 10 crianças. Ele entra umas 10h30, ele fica um pouquinho, ele dá o almoço.
Se a tarde, né, ele pegasse,
Hello?
Agora, bom, podemos voltar agora com a próxima apresentação. Tá, já, né? A próxima apresentação que nós vamos ter é sobre...
The next presentation will be about biosimilars interchangeability. And Cynthia, who is the General Safety Lead from Mexico, will speak with us. She is connected from Mexico.
Hello, everyone. Can you hear me?
Yes, we can hear you.
Perfect. Okay, so thank you very much for this invitation to all Jack Leckerman team. I feel very proud and grateful to be part of such an important and successful program. And thank you to Liliana Vargas and Germán Huertas and Jack Leckerman team for this invitation. I'm going to talk about biosimilars and interchangeability. So biologics have transformed the lives of millions of people. By reducing costs and creating choice, biosimilars increase the availability of these critical medicines to millions more. The interchangeability designation takes the concept one step further. As a catalyst for automatic substitution at the pharmacy level, interchangeable biosimilars may help drive efficient use of biosimilars and maximize the cost-saving potential of these important medicines. But what is a biosimilar? A biosimilar is a biologic product that is approved based on demonstrating that it's highly similar to an approved biologic product, known as a reference product, and has no clinical meaningful difference in terms of safety, effectiveness from reference product. We have some key points that we need to consider before to try to understand what is the interchangeability of biosimilars. A biosimilar is never going to be identical to its reference biological product. Therefore, biologics are not regulated as generics in the leading markets. the foundation of biosimilar is analytical similarity and to identify difference between a potential biosimilar and its reference product. The impact of any difference of this analytical similarity must generally be assessed in the clinic to determine if the difference matter. And each biosimilar developer may follow a different development program depending on product characteristics and manufacturing process. So here we could see the world turn upside down as we are working on safety. For example, for novel biologic, the main important is the clinical studies, and then clinical pharmacology, and then non-clinical studies, and then the analytics. The analytics is the last thing where we think when we are working with a novel biologic. But what happened with a biosimilar? We are based, the main base is the analytical test or analytical criteria. And then we are going to work, if we have some detections about analytical criteria, we're going to work with additional clinical studies. As I mentioned, the foundation of biosimilar is this analytical similarity to identify difference between a potential biosimilar and its reference product. This approval process includes a full profile of non-clinical and clinical data, and originator biologics must demonstrate safety and effectiveness in clinical trials to gain approval for each indication. So we have analytical studies to establish similarity between the biological and reference product, including minor difference in clinical inactive ingredients. Then we have the animal studies where we could see the evaluation of toxicity. And then clinical studies it also includes assessment of immunogenicity, PK, PD studies to ensure safety of the product in condition of use. The similarity in established between the biosimilar and the reference product is based in the totality of the evidence, including comparative, analytical. This contains structural, functional characterization, non-clinical evaluation, pharmacokinetic, exposure, safety data, and additional comparative studies. So this is going to treat like if it is biosimilar, we need to make a comparative analytical study. And then we have to go to a PK/PD non-clinical study for step 2. And then we could have, in order to have a strong evidence of biosimilarity, we could go to a stronger clinical data requirements. But what is the interchangeability? Biosimilar interchangeability. The definition is that biological product is similar to the reference product, can be expected to produce the same clinical result as the reference product in any given patient, and for product administered more than once, the safety and reduced efficacy risk of switching are not greater than with repeated use of the reference product without switching. So as summary, the definition is when we have two products, this can be exchanged one with another without a significant risk of an adverse health outcome. And we have two definitions that are going to be important of these interchangeability definitions. One is the automatic substitution, which is the practice by which the product other than the one specified on the prescription is dispensed to the patients without the prior informed consent to the treating physician. And then the switching, which is the decision of a physician to charge the patient from one drug to another drug with the same therapeutic intent in order to optimize therapy and reduce adverse effects. But what makes a biosimilar and interchangeable biosimilar different? First, to be designated as interchangeable biosimilar, the FDA or the health authority that we could consider as reference must require some evidence, some additional evidence in order to determine that could be an interchangeable biosimilar. All biosimilars are not interchangeables. So first, an interchangeable biosimilar must meet with the high standard of reference of a biosimilar. For example, they need to prove that they are highly similar structurally to the reference product and that they don't have a clinical meaningful difference in safety, potency, and purity. And this interchangeable designation requires an additional studies for a multiple substitution in patients. This is an interchangeable study. And this study shows how patients do when they are switched back and forth from a reference product to an interchangeable biosimilar. This study is designed to give providers confidence that the patients can be switched with no increased risk in terms of safety or that is going to change some terms of efficacy as compared to remain of the reference product. So this is the most important thing, that the interchangeable biosimilar products are proof that they are considered in the same way that has the same characteristics according safety, efficacy, and potency that biosimilar. So, but between these decision must be present an interchangeable study. So what is the interchangeable biosimilar? This meets the high standards of a biosimilar plus additional requirements, including one or more interchangeable studies. And you can expect the same clinical result as the reference product in any given patients. So this study, interchangeable study for this the intention is that demonstrate that there is no additional risk for the patients in order maybe that when they are under treatment for this biosimilar product, and there is no reduced efficacy when a patient is switched back and forth multiple times within different products and compared with the reference product. So this interchangeable biosimilar may be auto-substituted from the reference product by a pharmacist. So here are an example, or this table explains the interchangeable biosimilars include the benefits of biosimilar. The only difference is that we need to run an interchangeable study and conduct this study accordingly in order to demonstrate that the biosimilar could be an interchangeable product. And is similar to the biosimilar and the reference product. So how is an interchangeable study conducted? Here you can see an image where is placed the steps that you need to keep in order to guarantee that is running an interchangeability study conducted in a positive way. So first we have after a 14 weeks running period where all patients are given the reference product, patients are randomized to the treatment groups of the control group for a 16-week double-blind period. This is the first one, the first step. The treatment group is switched to the interchangeable biosimilar candidate. Then the control group remains on the reference product. And then after 4 weeks of the treatment, the group is switched back to the reference product. And after another 4 weeks, the treatment group is switched back to the interchangeable biosimilar candidate, and the control group remains on the reference product for 16 weeks. So here is very important to demonstrate that in all these interchangeable study, safety is a very critical part, and the tolerability is a very important part of this study. Also the efficacy, but we are going to consider safety, efficacy, and tolerability. So what does an interchangeability study show? Patients in treatment groups are compared to the control group to ensure there is no significant difference in PK, efficacy, safety, and adverse events. And why is an interchangeability study important? Because we required to prove that a biosimilar could achieve the interchangeability designation and characteristics. And they are designed to show that there is no increased risk about safety or loss of efficacy when patients are switched back and forth multiple times from the reference product to the interchangeable biosimilar. So interchangeability designation enables auto-substitution as pharmacy level. Once the health authority of reference has determined that a biosimilar has met the additional requirements to earn an interchangeable biosimilar designation, a pharmacist may auto-substitute that product for the reference product. And then individual state laws govern how and whether providers must be notified. The auto-substitution is available for both new and existing patients. Is there a concern about to receive an interchangeable product in place of the prescribed reference product? This is something that maybe we could have as patients some doubts about if there is any difference about safety or efficacy about an interchangeable product. And the response is that the interchangeable product will have the same clinical results as reference products, and the prescriber and their patients can assure that FDA or the agency that could approve has been proved this and has tested these drugs in order to meet high standards for approval. And the meeting standards means that the healthcare professionals and patients can be assured of the safety and effectiveness of an interchangeable product, just as they would be for a reference product. And what are the standards that the interchangeable products have to meet? Well, we have to guarantee that the patient, that they have efficacy, they have the safety guarantee, and that they have met all the requirements that are established by regulatory authorities that approve it. So is there a difference between receiving a reference product and a biosimilar product? There are no clinical meaningful difference between taking reference product and biosimilar. As reference product and biosimilar met requirements, FDA requirements or rigorous standards for approval for indications. And once a similar has been approved, patients and healthcare professionals can be assured of the safety and effectiveness of biosimilar just as they would for reference product. So one of the questions that we may have is that for interchangeability, biosimilars approved for all the same indications as reference product. And here is one very important point, that biosimilar products may be approved for all the same indications and reference product, but maybe we could have some products that only could be approved by a fewer indications. So for this, it's very important to go to the prescribing information and check the approved indication to determine what is the most appropriate product for the patient and their needs. So this is like a brief summary. Can a biosimilar interchangeable product be used in patients who have previously been treated with the reference product? Yes, biosimilar and interchangeable products can be used in patients who have previously been treated with reference product, treatment experience, or as well as patient naive or treatment naive. Before approval of the biosimilar product, FDA may request additional data that looks at safety information for treatment experienced patients who undergo a single transition from reference product to a biosimilar product. And as part for the approval process for interchangeable products, even more than once manufacturers must show that patients can be switched back and forth between the reference product. So this is very important in terms of safety and loss of efficacy. And here is the key in order for the implementation of these interchangeable studies, because we need to present this in order to demonstrate that a biosimilar could be interchangeable. So where can I find more information about a biosimilar and interchangeable products? Well, the FDA has a list of licensed biological products with reference product exclusivity and biosimilars of interchangeable evaluations. This is known as Purple Book. And this is an online resource for healthcare professionals and patients to locate information and identify information about what are the approved biological products. This Purple Book provides information about whether a biological product is a reference product, biosimilar, or interchangeable product. So this is going to be in the Drug FDA website. So this is a slide where I would like to explain in this slide what are the difference between the interchangeability and biosimilarity. And as you can see, the first is that the expectation is the same clinical result in any given patient. And for product that is administered more than once, there is no additional risk for safety or diminished efficacy as a result of switching. But this, again, is going to be completed and confirmed with these interchangeable studies. So the availability of biosimilar and interchangeable products that meet the FDA robust approval standards will improve access to biological products through lower treatment cost. So one of the important is that the patients could have different options to maintain their treatment. And this is a great opportunity to develop or to try to figure out if there is a different option for patients that could need, and a different way to demonstrate or use the drugs, also with all the efficacy and safety studies that must guarantee that the patient is taking a drug that could be useful for him, and also caring the patients. So thank you very much. And thank you again, Jack Leckerman, for the invitation.
Cynthia, thank you. Thank you so much. Hope you are able to hear people applauding you here. So we have one question from the chat here. Is COFEPRIS require any clinical trial, any studies regarding interchangeability, or COFEPRIS is not at that level yet?
Well, it's difficult because there is COFEPRIS, according to my experience, COFEPRIS is giving and trying to give this increase of to align this standard for ICH and this high level FDA and EMA standards. So I think, and I am sure that COFEPRIS could be working in these interchangeable studies, or to demonstrate that it could be an option for patients in Mexico. However, at this moment, there is no approved evidence or guideline where we could detect this possibility for interchangeable studies without a clinical studies demonstrated. So in Mexico, also, or we are in the health authority try to guarantee the safety and efficacy also with clinical studies. So for they are very important, the clinical studies, the results in order to guarantee the safety and efficacy. And it's the same that it's happened with safety and pharmacovigilance, that the health authority expects that we could have a clinical study and that we could expect that the real evidence we could have to try to have with a clinical study. But you can see with safety all the adverse information or the adverse event or safety information that you could have. So I think that the health authority is given like is given some advance in the way of working and the way of taking the guidelines and rules. But yes, at this moment is strong the need to demonstrate the information with clinical studies at this moment.
Thank you. Let me see if there is any other question. We have one question here.
Hi, this is Daniel Freire. I'm the medical director for Sandoz in Latin America. Many people, including myself, in Europe, have questioned the need for interchangeable designation as the FDA asks, because we believe that the quality standards for approval of a biosimilar and an interchangeable biosimilar are the same. The FDA says that the expectation of a patient and a healthcare profession when prescribing a biosimilar, being interchangeable or not, would be the same as the reference medicine, being for a naive patient or a patient already using a biologic. So my question would be, what is the scientific basis of this separate designation, considering that from a molecular standpoint, which is the one that drives the approval of a biosimilar, the biologic, the reference biologic and the biosimilar are indistinguishable products.
So your question, and hi, nice to see some colleagues from Mexico there in Brazil. So your question is that what is the difference, or what are the difference for the approval?
My question was, my question was, what would be the scientific basis of this differentiation? I understand that an additional clinical trial would bring the product a different designation, but from a scientific perspective, this could not make these two products different. This would be my question. People would not be more assured by a clinical trial showing showing interchangeability, then they would have to be assured about a biosimilar approved in a very stringent market like the US.
Yes, thank you. Yeah, this clinical or this scientific perspective or scientific rationale comes from the analytical proofs. So this rationale and for all this perspective to go for the biosimilar to an interchangeable product must be covered with two vias. The first one is the analytical studies, where we could have all the details about that we have to meet all the information criteria in the analytical studies. And then if we have more information or we need more information because we saw additional information or difference in analytical products between these two products, we could cover with an additional studies. But here the main point for this confirmation for an interchangeability product is first the analytical proofs, and the second one are the interchangeable studies that could be covered that during, as I mentioned in this presentation. The important thing here is that we could cover and demonstrate the tolerability, the safety, and the efficacy.
Okay, thank you. Thank you for the question, Neil. Thank you, Cynthia. And thank you so much for your presentation. That's very clear. We, at the end of next Friday, we share your presentation with the entire team here. So thank you so much to stay with us. So let's move to the next session. Alessio is going to be on the stage. The next session, it's about development of new antibody purification platforms. Okay, thank you for that.
Olá, boa tarde a todos. Primeiro, eu queria agradecer mais uma vez o
Hello,
espaço.
good afternoon. First of all, I'd like to thank this space in this event. This is my third edition following up this event to talk a little bit about biosimilars. And today I bring about how Cytiva, as a solution provider, is working to develop new antibody purification platforms and advance the existing ones. So I'm Alessio Pimenta. I'm product manager in downstream Latin America for Cytiva. So here you have my directions, alessio.pimenta@cytiva.com. Specifically for this area, antibody purification platforms. So we have very good presentation this morning by Merck talking about the intensification for upstream processes. So if we consider the upstream platforms in the last few years, we saw an intensification of process, which led to a significant increment on how much API we can produce in a bioreactor. So when we visualize traditional, conventional productive plants, there's always a bottleneck that was in upstream. We needed time to culture and low use on the downstream lines. We need the material to come from upstream to run the downstream production of proteins. But this scenario is changing. We've been decreasing the times of cultivation to obtain the same mass of product or even more. And now we are taking the bottleneck to downstream. And added to this increase of productivity in upstream, we had a change in our biodrug market when we see a demand on more targeted therapies with smaller lots of products. So we have families of molecules, but acting in different segments of disorders with a smaller number of patients, and the productive demand is lower and lower. So on one hand, I have the pressure of a productive, increasingly productive upstream, market demanding productivity from our productive plants, and a downstream line that has to be improved to respond to this demand. So increase productivity and flexibility so we can have different monoclonal antibodies, biosimilar or reference in the same platform where we will use the same setting of unit of a single production, but I will be able to exchange molecules in larger campaigns throughout the year. So these are the main challenges for the company currently that present us, which are our goal for development. So translating that to downstream, how can we translate in goals to improve our downstream technology? Well, first consideration is the risk of contamination due to bioburden. So in order to achieve flexibility and work with different molecules, I need a very robust sanitization or a completely single-use productive line. In addition, due to the different upstream times, increase the antibody capture capacity, which are the first processes of purification of a monoclonal. So I have to work faster and be able to capture this huge amount of antibody that upstream is generating. And as I said, work with flexibility to increase the multi-use of a same productive line. So in addition to help us in productivity, this also provides tools so the producers of the molecules have a fast time to market at a lower cost to respond to price requirements that biosimilar put in our market, in biotechnology market. So how can we overcome these challenges and bring platforms that are ready to use and depend on just a little development, like a toolbox that you can immediately apply in your process. So recently we introduced to the market a new platform for antibody, a new resin, for antibody capture, is based on protein A capture, is MAB Select Prisma A, and the advancements of this resin are high resistant to sanitation products, which helps with contamination risk. It works with concentration 10 times more than the resins in the market for sanitizing chromatographic columns. And in addition, it has a high productivity and a capture potential that are much better than traditional resins. So, as for bioburden, why is this so important to work in a safer environment, especially in multiple purpose plants? We have here an extrapolation on how much a cost of a contamination may achieve, considering the moment we identify the contamination in our process. We can, it varies from $20,000 if it is identified in the very beginning in quality control, going to sanitation of the facility, or even exchange or replace resin that can go up to $100,000 if you lose a complete chromatographic column. But the worst of all, if you already have a lot ready for commercialization and you discover a contamination and you have to dispose a whole lot, it may achieve $1 million. So imagine the risk that this implies. And the focus of contamination currently is in downstream, because we have upstream as a source of contamination. We have culture media there to develop bacteria, to propagate contamination. However, as we have a very well-established single-use process in the upstream, we have a good mitigation of this risk working with disposable bags. Whereas downstream is an open loop, is an open operation, and is not sterile. So although sanitized, is not sterile. So we have to focus on contamination control. And what do we do with this resin? This family, MAB Select, is based in A protein. So we have agarose matrix conjugated to protein A ligand, which is responsible to interact with FC receptor of the antibody, capturing the antibody in the resin. So we have a clarification process right after upstream to remove cellular debris that comes from a bioreactor, which is the supernatant of my bioreactor. And this supernatant is rejected in a chromatographic column. So the role of the resin is captured so we can concentrate and follow to the purification. So this protein A ligand was modified with recombinant RNA technology. So we identified the main residues that jeopardize the stability of the protein, decreasing with a high concentrated sodium hydroxide solution. And with that, we have a ligand that is much more resistant to sodium hydroxide. So to the blue line in the chart is how much antibody each ml of resin can capture after 50, 100, up to 300 cycles of sanitization with sodium hydroxide. So in blue, we have the new MAB Select Prisma, and green MAB Select XL, and orange MAB Select Sure. So 3 generations of the same resin family. And the test was with 0.5 mole of sodium hydroxide. And my new ligand maintained up to more than 93% of their capacity after 300 cycles with 0.5 molar sodium hydroxide. The others is less than 55, less than 40. So we increase a lot of the use of the column. If we compare to the others, more than 50% of the binding capacity comparing with the others. In addition to this study with 0.5 molar, I will show you a study with 1 molar of NaOH. So this was to solve the issue of contamination, improve protein ligand to be more resistant to higher sodium hydroxide concentrations, because with that I increase the time of life of my column, and this provides more safety to my process.
Besides that, we also changed the structure of the resin. The high flow matrix, which is the base to use the ligand, also has some different qualities. Its diameter is a bit lower, while traditional ones are around 85 micrometers. This one has 60 micrometers, and it has wider pores, meaning that the mass transfer from your product to the ligand is much more efficient and faster. So the first graph here on the left shows us the dynamic binding capacity of the 3 resin generations due to residence time, which is the time we allow the product to permeate inside the column, interacting with a larger surface of resin. So here we see the residence time, 6 minutes, and there for Prisma it's 80 grams per liter, 60 grams per liter for sure at LX, and 50 for sure. So a significant increase again in the productivity of these resins. And on the right, the graph I had mentioned with a 1 sodium molar. So 90% of its binding capacity remains after about 15 minutes contact time per cycle, which is great and reduces the number or reduces the odds of having contamination. So you can increase the number of usage cycles for that resin, and you can have more productive resins. But how does that benefit your process? Here's a graph showing the normalized cost per capture batch. So how does that translate? For Prisma, we can work with lower resin volumes, and we could also use that lower volume for a higher number of cycles. So it's an expected cost reduction of 75% in a unit operation. If you've worked in that monoclonal application process, you know that one of the highest costs is capturing antibodies because of the cost of the resin. So now being able to develop a new unit operation where you start with a cost of 25% of the current ones is a significant gain for the industry. However, complying with that solves the issue of contamination and the need for protein A, but it doesn't create the flexibility that we expect downstream. Chromatographic resin is not single use. In order to reach 100% of our investment, and remember, we saw 150 cycles. What good is it to have a 100% single use upstream where you discard your consumables batch by batch, while downstream, although you have single use equipment, and right now we're ready to use 100% single use equipment for chromatography, where the entire material that comes in contact with the product is removed batch by batch, just as we do with single use, but you still have that bottleneck of the column. Of course, you can invest in 200 liters of resin, but it will only be used in a single batch and then discarded. And the cost of that is quite high. So that's the main point today. That's the main bottleneck to make the downstream process 100% single use. And how are we addressing this issue? Cytiva acquired a startup a number of years ago, and we developed a chromatographic device based on cellulose fibers. So this device here in the middle, called Fibro, represents this new device. So what are the difference between fibro, resin, and chromatographic membranes or monoliths that are already available in the market? Resins give us the highest interaction surface. So here we see EM images at the same scale. So you can see that the interaction surface is much higher. The productivity of a resin is much better, but its process is lower, as we'll see later on. Membranes give us a good purification process because you don't have pores, but rather channels. And you can see a lot of empty spaces within the membranes. The mass transfer is direct. 100% of the capacity is reached very quickly, but the surface area is very limited. It's 20 times lower than a resin. So normally membrane applications are more specific for viruses, for molecules that have a higher level of need, and fibers occupy the space between these two universes. So by the surface it has, you still have channels, but the diameter of the channels is much lower than the membrane. It doesn't necessarily need pore diffusion, so you still have direct interaction, but you can increase your surface area considerably. So we're trying to find a device that has the best of both worlds. And now we have some products in the market. This is the high trap and high screen lines. They're in development, and they will develop two industrial products. They'll be very similar to the tangential filtration cartridges, which are flat sheets. Here, you're going to have devices in different volumes at a pilot scale with large-scale productivity. This line is completely compatible with chromatographers in the market, single-use or traditional. So developing this new technology does not mean that we will need to change the hard risk structure in your production plan. It's a matter of adapting your device. And this shows the differences in what we gain from this device. So in a resin, we have convective flow, the flow through which your product goes to the column and how it produces through its pumps. And you need to have enough binding for your product not to only interact with the surface of the resin and the matrix. It needs to penetrate the pores and diffuse. So there are two physical processes, a convective flow and a pore diffusion into the pores of your resin to access 100% of the ligands you have available. So this is what we call residence time. Having residence time too low means that your product is not reaching 100% of the ligands in a resin, meaning that you're wasting money. You need to have higher resin volumes for a lower percentage of the product that is not reaching the center of your resin. So this is how it works. When you use chromatographic resins, you have high productivity processes that take a long time, that are slow. With fibro and with membranes, since you do not have the pores, since mass transfer is direct, residence time here is basically seconds. It's almost instantaneous. So you don't need to be concerned, and you can work with much faster processes and flows reaching 100% of your available ligands. So when we analyze how that will impact our process, the graph here on the left shows residence time in seconds and your binding capacity. The blue curve shows how the resin behaves with its residence time. The higher it is, the more the product will penetrate the pores and the higher your dynamic capacity. So you need a scale of a few minutes to reach a good binding capacity in your chromatographic resin. With fibro, you can immediately reach its potential. So in this scale of time, in seconds, fibro is going to be more productive than a chromatographic resin. Of course, if you have more residence time, it will go up and its capacity will be higher. To the right, we see a full chromatogram showing the capture of monoclonal antibodies with a fibro. And you can see here the scale. It's a full chromatogram in 4 minutes and not in 4 hours as the traditional process goes. So it's an exponential gain of time when you compare a fibro to a chromatographic column. Here are some studies showing the quality of the device when it comes to its resin. So this first one shows different antibodies purified. In gray, we see bispecific antibodies, and in blue, fused antibodies. So the ligand in the fibro is the same as in the resin. So its purification should be similar. The device is changing, but the ligand will be exactly the same. It's the same protein from the resin. So here we can see that the variability of molecules that it can capture is the same as the resin. So there are no losses in flexibility of the ligand in this kind of device. This is the quality of the product that we are purifying in recovery, aggregate level, leak, and HCPs. So in blue, we see fibro, and in green, the traditional resin. So in some parameters, it's actually better on process control parameters. And this is the same sanitization study showing that the same behavior that the ligand has in resin, the same resistance is also here in the fibro device. So 300 cycles with different concentrations from 0.5 molar to 2 molar, the same ligation capacity or the same binding capacity as in the resin. So the initial goals that we had with the first resin also apply to this ligand in the fibro unit. Next slide, please. These are a few studies with reproducibility. So in this study, we have a prototype, 5 ml, its performance for 50 cycles. And on the right, we see the curves showing no perceptible variations. So the black line here is absorbance at 280 antibody, and here we have 50 lines superimposed, showing that there's no difference in the results obtained in these 50 cycles. And the graph on the right shows what happens to the elution peak area and the delta pressure over cycles. And we'll see at the end how we're going to use these parameters and these properties of the drug to reach single-use chromatography. This is the same study, but with a different scale. The scale is higher. So we use 17 cycles here. All of them overlapped and, superimposed in the blue line with no significant difference. And the production device, 2.4 liters. So the aim here was to show the properties of sanitization, short process times, and reproducibility at a bench and at an industrial scale. So now I have a device that can be adjusted according to the chromatography, working with a fast cycle in comparison to a chromatographic column. So how can I make use of these advantages from the device to try to design a single-use chromatography? If I have a bioreactor and I need to recapture, what is the industry design now? We need a chromatographic column that has a volume of 60, 55 liters of resin to capture the protein generated in these liters at once in a single pass, to go through the column and continue with polishing. This process will take 4 to 24 hours, depending on your process. It can be, injection stage, 20, 25 hours. And you're going to need to work with this column through 300 cycles. Now, imagine if you have a plant that is still being approved, and if you need 10 batches a year, or if you want to use different products, and you have this limitation where you cannot use this different antibodies in the same column, you'll need to keep it for 300 cycles, and that can take 15 years depending on your demand. So what do we propose with this device? Instead of designing a device that can capture 2,000 liters at once, I can make use of the fact that the process time is minutes long and do, for example, 200 cycles to process the same 2,000 liters. So you can take a fibro device that is 2/100 of the volume you need and perform it 200 times. This 200 cycles in the same budget and accumulate. And at the end, your process will have been faster than the chromatography column, and you'll have used that 200 cycles that resin can offer. So at the end, you'll be able to take this device and throw it away, just like we do with a single-use bag or bioreactor. So this is what we're trying to do. With this kind of device, we want to expand the available ligands and have a downstream chromatography stage that is 100% single-use, meaning that all the material that will contact your material will be discarded batch by batch. Will that replace resins? No. So here we are expanding our possible applications, and Fibro will be designed for multi-product facilities who will work with smaller production volumes. So we're going to have a new plant for biosimilars. Let's imagine that I want its capacity to produce biosimilars to be higher. So you're going to have a smaller bioreactor, and up to this volume, we can create a capture stage with fibro, and it will be more economically advantageous. But if you have a traditional plan that is dedicated to a single monoclonal antibody, and that's all you're doing the entire year, then a resin might be more applicable. So we're adding a new technology. Our intention is not to replace high productivity resins with this product. So this demonstrates a case study from Sanofi. They tested this device. They had two studies. One was a reproducibility study with and without sanitization. So this is another advantage that we can explore in this kind of device. Since it will be discarded after a batch, it means that you can execute 100 to 200 cycles without having to wash between samples. So that saves time. And on the left-hand side graph, you can see 75 cycles without any washing between the different sample injections. And the second graph, or the second study, shows the sanitization for 5 sample cycles. So from no sanitization to full sanitization. And when we receive the maximum working pressure, it means that you can no longer inject a sample because it's starting to clog. And with that, they got 75 cycles. But if you sanitize with every 5 cycles, then you can reach 200 cycles until maximum pressure is reached. And this is the scale-up process. They use these results from cleaning every 5 cycles to test different volumes. So from a 4 ml scale to a pilot scale of 40 ml with a productivity of 436 grams per liter per hour capture from this device without risk of contamination or degradation of the final product quality. Well, these are my takeaways. First, the company is focused on working on technologies that can be that will demand less development. So platforms that have been tested in-house that can be provided to you pre-developed. So with just some small adjustments for your products. We use a change in our A ligand protein to increase resistance to sanitization. We change the structure in the matrix to increase productivity and to have a resin that will help us solve this bottleneck in the downstream process due to the high upstream titers, and to provide flexibility to a 100% single-use plant. We are developing this kind of disposable device for chromatography. So this is an example of how we work, our toolbox. So if you come to our website or talk to our team, we have a number of suggestions that can be tested together for different kinds of molecules. So if you need a bispecific antibody, there's a pre-process that can be provided. And instead of saying, that we have 60 different resins and ask which one you want to use, you want to be able to facilitate your work and provide a ready-to-use solution. And if you're interested in finding out more about how much this can be made into savings for your process, we have an online tool, ctiva.com/process intensifier, where you can input some data and simulate what would happen to your productivity and your costs if you switch to MabSelect Prisma or Fiber. We have an online tool to help you with that, to see if that is a worthy investment for you, and to see, what financial impact these changes will have to your process. That's all. Thank you, everyone. Thank you, Alessio. Do we have any questions for Alessio, or can we continue with the next session? I think because of time, we can leave the questions for our break so that we can continue with our program. Okay. Now we will hear from Diego Rodriguez, who is a consultant in strategic innovation. He is in Colombia, and he's going to tell us a bit about biosimilars market innovation, from developing a mindset for global competitiveness to business strategies. I think we're ready, right, Diego? Welcome.
Good afternoon, everyone. Can you hear me? Yeah. So allow me to share my screen. Can you see it? Yes. Well, first of all, thank you very much for inviting me for this event. This is the second time I take part in the Biosimilar LATAM, which is a privilege to be with you and be part of this panel among all the talks today that were very interesting. So I will talk about a topic that is the innovation in biosimilar market, and how can we develop and how can we change mindset to achieve global competitiveness and translate it into business strategy in biosimilar market. I've worked in biosimilar and reference drug market, both in innovation and processes. And this allowed me to try some of the things I will address today. So it is not just theoretically, but practical experience. So I will start with a concept that maybe is very clear for many of you in this session, what is an innovative medicine? I know that it's very clear for you. We discussed this in the previous presentation as well, but we didn't discuss what is a biosimilar. When I say that is a biosimilar, in a pharmacology book, there is something that really calls my attention, is that biosimilar or similar drugs or non-innovative or copy products. So means this is my first doubt. So if I say that a biosimilar or similar is a copy, then it is completely the opposite as innovation. So a biosimilar cannot be innovative because according the terminology, according pharmacology literature, these are completely opposite things. And this is what I will discuss in this session. So we will consider, first of all, what is innovation? What you see right now in the screen, when we say the word innovation and innovation definitions, we see that one of the definitions is creation. That is maybe one of the most solid concepts, but we also have modification, movement, alteration. So maybe innovation is not as strict as we may think. And that's why today we will see the different traits of innovation, and this will prove how biosimilar can be innovative. So we have one that is radical or disruptive, and the other type of innovation is incremental. I will discuss this into depth. And we can also innovate not only in the science of the product, but also in the business model, which is the case in the biosimilar market. So when we have a radical or disruptive innovation is when we had, for instance, we had here the popular BlackBerry. I believe most of you in the session had one once. That was the maximum behavior of a mobile back that time, with a keyboard to operate the device. And then Steve Jobs made the major disruption that keyboard is no longer necessary. So this leads to a radical, disruptive change, because changed everything we had in hand. And the concept I have for the device is now completely different, because I can, in place of 40 keys of a keyboard, I have just one, and I can do everything. So this is a radical or disruptive innovation. So, but there is an incremental, the incremental type, the incremental innovation. I want to illustrate with an example. Colgate, the dental cream, reached the market, but they want to sell more. So sales executives of the company got together to discuss how can we, or what can we do to sell more. So in marketing, we have to generate new users, and this is an option to sell more. Is one alternative. Second, increase the size of market, but they were already leader, already well positioned, so this no longer used. So I have to, we have to develop new ideas, so we find a more efficient solution. And in this case, an employee said, I had a very nice idea that to sell more dental cream, we increase the size of the nozzle from 1 to 3 millimeters, so that in diameter. So everyone, when using, will use more, and this will end up in more sales. So this is incremental, because it's the same product. It's just one single modification, an increase in diameter that leads to more sales. So this type of innovation are all innovation, but in different ways. And the second part, we have innovation in the product level. And here, specifically in the pharmaceutical sector, pharmaceutical industry, when we have a molecule and we have now a different device. So we are not changing the API, but we are changing the product, because this new device may be less painful to apply, or more efficient, or reduce adverse events, and so on and so forth. So this product innovation is not necessarily changing the base product, but the way we can complement. So we have a differential in the product, and this is also innovation. And another type is business model. And then we talk about business innovation. And one of the most clear examples is Airbnb, Uber, when we see that this is a non-conventional business model, because for the other type, we have to invest a lot of capital to a new business. And Airbnb do, well, what we have of common use, we transform in business when it's not being used, like a room in your house. So we've, in a very easy way, we see a range of possibilities of innovation. And although they are all different scenarios, they all lead to innovation. And when we apply it to biosimilars, it's even more interesting. So when I see that, and remembering the first slide where we said, an innovative product or reference product is maybe the greatest innovation of a pharma market and pharma industry, and biosimilar is a copy. So it's a bioequivalent, but it's not really a copy. It can be innovation if we add the 4 areas we discussed in the previous slide. So with that, a first message I have: in this market, we have a great opportunity with less investment or risk, because in the end of the day, when we are biosimilar, we know that there is a lab that already investigated, conducted clinical trials, developed the product, and somehow opened the market, creating positioning among healthcare professionals, and we don't have to do it. The investment of that is done. So we just have to innovate to differentiate our product from the reference. So, and then we can apply the 4 areas we saw in this previous slide. Radical, disruptor, incremental product or business. And now, well, what can we think to innovate? So considering that the pharmaceutical market presents a lot of limitations from the regulatory standpoint. So first of all, we have to use locally innovation techniques as global companies do. I know that most of large companies in this session are multinational, but when we talk about other global, I mean also other industries, TR, IT, textile industry, and any other industry. And in this case, there are techniques that are known like design thinking that allow from any product and from any industry, we can have a business idea that is really connected to the customer need. If we give these 5 steps, this is very important in the brand plan development for pharma industry. You can have a brand plan. So we follow these 5 steps, and this may lead to a solution that is related to what the customer needs, that is technically feasible and profitable in the economic from the economic standpoint, because we are a corporation and we work for profit. And many of you probably know that when executing, we can interact in a more simple way and achieve good results. So step number 1, use these techniques used by major global companies, but in our local market, regardless the country in Latin America you are. And second, motivate innovation from every area. And here we see examples of the most innovative countries in the world. Sweden, 20 years ago, was in a very complex situation, and they decided to create the model, triple Alex model, that gathers industry, state, or governmental authorities and academia. When they all get together and work for innovation, they can achieve much more assertive and innovative ideas that cannot be created when you are working in isolation, when you're working by yourself. So in the case of a pharma industry, they developed a project with a local university. In the case of Brazil, for instance, you can gather a governmental agency. In the case of Colombia, we are incorporating governmental bodies that are connected to the area of entrepreneurship and innovation, and companies that are eager to innovate. So when we have that, you can give a step forward, because when executing the innovative ideas, you have business feasibility that was previously approved because you included the approval stakeholders for the scenario. When we talk about the state is developing a project with regulating authorities already from the beginning of the project. This enable ideas and reduce approval steps. And the third point, maybe one of the most important ones, is working with synetics. When I combine 2 things. So, for instance, when I use a wheel that I had in a carriage with horses, and now I connect this to an engine, I can have a car. But when Ford designed it, it was not, there's a sentence he used to say, if he asked what people wanted, maybe people would say, well, faster horses. No one will say a car with internal combustion engine. So we, pharma industry, we focus sometimes too much in hospital, clinics, and logistic distributors, but there are other sectors where we can be involved and partner with companies that in the first moment may sound of having no sense, but will add. So we already have the conclusion that biosimilar can be an innovation when we have tools helping us to innovate. So now let's see the 4 basic stages of product flow, so we know how to tackle innovation. So first of all is product development. In product development, the base probably is molecule, but let's imagine we already have the molecule. So we can innovate with a device, or the administration mode, or the portfolio of the product. And in the portfolio, an additional analysis here. And actually, when I worked in a biosimilar and equivalent equipment, the portfolio is too technical, talking about biology and endocrinology and the level. But when we talk about portfolio, is, well, if metaverse exists is enough, and artificial intelligence is enough, so why don't I put my portfolio that? Or why not following up my product with artificial intelligence? So these are the possibilities that we can develop in this scenario. Another point is, I believe that the previous talk, we were discussing a little bit about this products. And the last point is this of technology, how to add all of that technology, artificial intelligence, metaverse, and all the emerging technologies. Well, but then we have the product in development, and so we go to the next stage, how to use that, how to launch, and then we have to access the regulatory authorities. If I include agencies from the beginning, and with that I have the dossier to present them, I have an additional step, and I can achieve things that usually is not seen from the standpoint of pharma industry. And the second is the distribution model. Here I'm considering distributors, wholesalers, retailers, because maybe we can develop a better business model that allows us to achieve farther distances or where our distributors do not get. So when we have all of that, we have to go to market. And here we have a very interesting, important. Education has to continue. And continual medical education is a scientifical. We are used to sessions where we sit and there's speakers and round tables. And as we can see in this forum, it can be virtual, in person, or hybrid. But we have to develop something different. We have to seek tactics or games that are used by children in school and deconstruct it. So when we present a concept is in a more robust fashion. So this is a way to innovate in continued medical education. And this can also be done in the access to governments and to authorities and agencies, because I can reach the hospital or clinic in a faster way. In terms of promotion, we have to highlight here alliances, and I will address this later on. For instance, not only, I cannot promote from the medical standpoint, I can, or virtual visit or calls. There are other ways like social media where we don't see product promotion. We could try to see how promotion start to evolve, and in a commercial level, how to produce new risk models, negotiation models that optimize our streams. And last, when the product is in the market, it has to reach the end consumer, that is the patient. So we have to evolve with the patient program and evolve them and co-create with them. And hence, we will have something that they really need, because they are the ones to consume the medications and pharmaceutical solutions that we develop in our companies. So the examples of each of those, for instance, in product development, there is a company that since 2017, they put in children, they apply vaccine shots, but when they are receiving the shot, they are connected to augmented reality glasses. And they put on the goggles, and with that, they are distracted, the pain is reduced. So I reiterate the example. Here, I can develop a different product. It is the same API, it is the same vaccine, but I have an exclusive device that can differentiate my product by decreasing the pain. This is a product, second example, from a company, Aflac, that developed this duck. What does the duck do? This is a duck for pediatric cancer patients that pass through painful treatment, and they simulate the pain sensation that the child may be feeling. And there is a circle in the duck's chest, and the children, they identify themselves with the duck, and this creates empathy, decreasing the anxiety of the children and the fear they have of the different procedures. And the image to the left, we see, or to the right, I'm sorry, the different injections and the empathy to the duck. Another example by Pfizer. It is a game that is directed with these drawings that it's deconstructive. So they have to deconstruct the scenarios, but the scenario is a scenario in a hospital. And what they try to do is recognize the scenarios of a hospital. So when they go there to receive their oncological treatment, it is more familiar and less traumatic in real life. So Pfizer hired a game company to differentiate their product and benefit the patients. Another example, that is a metaverse platform called the Decentraland, that is a universe where they have a sector that is called Big Pharma. And what does Big Pharma do? Through artificial intelligence, they simulate chemicals. And if you want to simulate any molecule, you go to Metaverse, you can use their plant and simulate the processes and observe what the result of the molecule could be. Well, I don't really know how effective is this, but what we can see in this scenario is that through Metaverse, we can reduce the risk that this molecule would generate in a first impact, because we have a simulation scenario where we can use artificial intelligence without having to invest so much upfront. We have another case that is Carbon Health, which is an app where we have all the basic healthcare services like diagnosis, exams, consultations, and it's a system that is very similar to, and when you walk 5 kilometers, you receive a point. You take a consultation, you send the exams, and you receive a specific points and you continue the process in a virtual platform. What can we see here? If are active, we are included somehow in this type of platform that can have better results.
In pre-launch, and as I was saying in previous slides, in Colombia, there's a company called Rappi that is also in Brazil that delivers different orders that you might make, from takeout to pharmacy, drugs, and so on. So they have had an alliance to deliver vaccines to people through Rappi since 2020. So I'm not sure what impact they had, but we can see a clear example of connecting two companies that in theory don't have anything to do with each other, but if it generates an innovative idea, they do it like this. And there's a great company in healthcare and as a game vendor called Tencent that joined Merck to create a platform with all health services in the same platform. So you can have food delivery, medication deliveries, consultations, and absolutely everything. And it's a metaverse scenario, so you can see how access is being provided, and it's omni-channel. So the entire healthcare system can be accessed through a company or a lab, focusing on a new technology, the metaverse. Now we have a couple of examples of going to market. This is an article on artificial intelligence, which is already diagnosing cancer very accurately, especially in imaging. And this is a reflection that we can have. We can see how artificial intelligence is advancing. Of course, I don't believe that it will ever replace a human physician, but if they're now diagnosing, probably in the future they will start making suggestions for people to be treated by a doctor. So this is a reflection that I'd just like to bring. Are biosimilars aligned to these artificial intelligence platforms? And can we have the best position possible as we do with physicians, of course, based on evidence and all the strategies that are used in treating different conditions? This is another example from Spain, a hospital that already has a virtual platform where you can go to the hospital, talk to a physician, and it is all done through a metaverse environment, which simulates the physical hospital. Finally, on the patient side, we have, for example, a challenge from Novartis called Beating Innovation. So it searches for healthcare solutions without replacing, of course, the medical community. It is basically a challenge that invited people to solve a specific issue, in this case, a cardiovascular disease, and ask people how it can be solved. So it allowed anyone to participate and maybe generate ideas that we would never have because of our own paradigms or because of what we already have in mind. So we can see how solution can be developed by patients. This is another example from AstraZeneca that had the first innovation hub in the region in Colombia. AstraZeneca wants to include that concept into their way of working, and that's how they're starting to work with different universities and entities in Colombia to create innovation, just as they were doing in Peru. So when you include different areas of society, it eventually generates innovation. So this is what they were attempting to do. And finally, there's this company called Presenis that, through one of the companies, created Expression Days, where basically they created artwork and painting to show how they were feeling. And these artworks are exhibited and receive awards.
Hacer algo de manera innovadora y involucrarlos dentro del proceso.
Entonces, ya cuando vemos todos estos escenarios y todos estos ejemplos, la pregunta es, ¿por qué no hacer esto en nuestros biosimilares? Si de todos los que yo les conté, básicamente la gran mayoría, todos están alineados a productos de referencia. Pero si nosotros, desde nuestro mercado biosimilar, nos colocamos el objetivo de generar innovación y no solo enfocarlo a que somos más acceso, podemos llegar a estas ideas innovadoras. Entonces, esos mensajes que invitamos que puedan llevar a casa es el primero, y es que no esperemos que la innovación llegue específicamente de esas casas matrices, de los headquarters, porque finalmente sabemos que estas compañías grandes tienen departamentos muy grandes a nivel global y básicamente lo que nos llega usualmente ya son las cosas que debemos ajustar a nuestros mercados. Pero, ¿por qué no crear alguna idea de estas? ¿Por qué no generar un mecanismo, un dispositivo, una forma de administración diferente, una realidad aumentada, realidad virtual, un metaverso, en donde desde nuestros países pueda subir hacia la región y de esta manera podamos tener un impacto no de arriba hacia abajo, sino de abajo hacia arriba? Pero para poder llegar a esto necesitamos invertir en el soporte, en el apoyo que podemos tener de conceptos de innovación para poderlo hacer de la manera adecuada, porque a veces cuando lo hacemos también sin experiencia nos puede tomar mucho más tiempo que cuando tenemos a alguien que nos pueda guiar. Segundo, es que si queremos tener una innovación de tipo estratégico o un soporte estratégico, debe ser apoyado desde todas las áreas. Muchas veces cuando estamos en equipos de liderazgo, tratamos de hacer todas las ideas desde arriba y luego cascadearlas hacia las demás personas de la compañía. Pero con el ejemplo, por ejemplo, que les daba de Colgate, nos damos cuenta cómo usted se involucra a las personas que están en Customer Facing todos los días, probablemente puede llegar a tener una idea de mayor valor y más directamente relacionada con lo que el cliente necesita, porque obviamente nosotros estábamos en la oficina usualmente, tenemos cifras de mercado, salimos eventualmente a campo, pero realmente las personas que están todo el día de cara con el cliente son las que realmente nos pueden traer una información de mayor valor. De hecho, esto en innovación lo llaman la TIC Data, esa data gruesa. La data gruesa nunca viene de arriba. La data gruesa viene de la persona que está en contacto todo el día con el cliente y de esta manera pues crear ideas aún más innovadoras y más eficientes, porque nosotros en el mercado de los biosimilares o bioequivalentes no solo somos más accesos, tenemos muchas más cosas que podemos aportar. Creemos oportunidades con innovación abierta y no solo con innovación cerrada. Cuando ya logremos generar una cultura organizacional en donde realmente se vive en la innovación y la forma para saber si se está viviendo la información es que al menos haya una persona liderando el proceso de innovación. Si en su compañía no hay una persona enfocada o apoyando los procesos de innovación, realmente usted solamente quiere innovar o quiere implementar innovación, pero no lo está haciendo. Pero retomando este punto que les estoy hablando eso se llama innovación cerrada, cuando usted solamente tiene involucradas a las personas de la compañía. Pero cuando usted también hace una innovación abierta, que es el ejemplo, por ejemplo, que les tocaba de Novartis o que les tocaba de AstraZeneca con el Tríplice Hélice, usted va a llegar a oportunidades aún más atractivas para el mercado. Entonces, aquí la invitación es no solo cree oportunidades desde la innovación cerrada, sino créela e incentive la innovación abierta. Motivar al ensayo, error, a lo intento y tengo errores y mejor, porque finalmente a veces, y en las compañías obviamente nos piden resultados financieros, crecimientos y demás, debemos romper el paradigma de perfección, porque finalmente, de hecho, hay una frase que dice que si usted no se está equivocando, es una seña de que usted no está innovando, porque finalmente cuando usted está innovando se está metiendo en un mercado que no sabe, sea porque sea una innovación incremental o sea porque sea una innovación disruptiva. Usted está llegando a un segmento que no sabe o a un escenario que todavía no conoce. Entonces, lo normal es que usted ahí tenga prueba-error, ¿sí? O tenga ensayo-error. Entonces, rompamos ese paradigma de perfección de creer que necesitamos en todo lo que intentamos ser exitosos y ganar, porque finalmente a veces cuando perdemos es cuando eso nos lleva a una idea que genera un impacto alto. Por otro punto, no esperemos llegar a una innovación de tipo disruptivo cuando no hemos hecho una innovación incremental. Obviamente, en el caso, el ejemplo que tocaba de los smartphones, pues finalmente pues fue muy chévere cuando Steve Jobs llega a este punto y llega a sacar el iPhone con una innovación totalmente disruptiva. Pero para llegar allá, él tuvo que tener una innovación de tipo incremental. Entonces, si nosotros no estamos generando dentro de nuestra compañía innovaciones incrementales, innovaciones que están mejorando nuestro producto, que están haciendo un diferencial en nuestro producto, pero no cambia el modelo de negocio, no cambia la forma del producto, pues finalmente hasta que no logremos esa innovación incremental, no vamos a poder tener un tipo disruptivo. Entonces, si usted quiere llegar a tener una idea súper innovadora, tiene que empezar con otro tipo de ideas que van a ayudar a la compañía a madurar en esos procesos de innovación. Hay otro punto importante que principalmente pasa en mercadeo y es refocalicemos esos A&P más hacia la innovación y medios hacia la estructura o al gasto tradicional. Entonces, por ejemplo, lo que les decía de la educación médica continuada. Obviamente sabemos que la educación médica continuada, que los médicos, obviamente hay un protocolo de alguna manera u otra para poder llegar a estos, pero a medida que nosotros empecemos a involucrar diferentes agentes, nos vamos a dar cuenta que lo podemos hacer de una manera diferente. Y cuando ya tengamos eso tan claro, empecemos a refocalizar A&P. Es más, ¿por qué no dejar, digamos, las personas que son líderes de mercadeo o líderes de unidades de negocio, ¿por qué no dejar una parte del A&P solamente para hacer prueba-error? Y de esta manera podamos empezar a llevarlos a una mayor innovación. Y si usted ve que de pronto a veces como que no fluyen mucho las ideas, haga un benchmarking, pero no lo haga solamente con la industria farmacéutica, sino con las otras compañías, por ejemplo, de tecnología, que son tal vez las que más en auge están en el tema de innovación. Genere portafolios robustos en sus áreas de interés y expanda o crezca acorde a la tendencia. Y en este caso hablamos específicamente de lo que les decía puntos anteriores, y es no solo nos focalicemos y rompamos el paradigma de que mi portafolio se amplía con más productos farmacéuticos, sino yo puedo ampliar mis productos, incluyendo tecnologías, incluyendo metaverso, incluyendo inteligencia artificial y todo lo demás. Y por último, nosotros podamos llegar a ser tan innovadores o más innovadores que el producto de referencia. ¿Por qué? Porque tenemos un abanico amplio para innovar. ¿Cuál es el punto? Que cambiemos ese mindset, tengamos claro que la innovación se da desde una mejora continua y de esta manera podemos llegar a diferenciarnos en nuestros mercados y, como les decía puntos anteriores, no solo enfocarnos que somos más acceso, sino somos más acceso, somos más innovadores y generamos un mejor producto para cada uno de los pacientes de nuestros mercados. Entonces, ya para cerrar, la pregunta es, ¿por qué no hacer mi biosimilar un producto innovador? Muchas gracias.
Obrigado. Alguém tem alguma pergunta para fazer? Eu creio que não. Diego, gracias por la presentación, gracias por su tiempo acá con nosotros y vamos a seguir acá con la agenda. Bien.
Muchas gracias.
Eu queria só fechar com 2 ou 3 pontos aqui rapidinho do final da nossa agenda de hoje. Acho que está todo mundo com o celular, né? Vocês poderiam só conectar nesse QR Code que a gente tem aqui, o mesmo QR Code que está no seu crachá. Eu pediria para vocês abrirem, a hora que vocês conectarem, vocês vão abrir com uma agenda, que é a agenda do evento. E tem uma parte azul que é o formulário da avaliação do evento. Eu pediria para vocês preencher, não leva mais que 1 minuto para a gente poder finalizar o dia. Se alguém tiver algum problema, é na primeira página. Bem na primeira página tem embaixo aí está em azul. Evaluation Form. Se vocês puderem preencher para a gente. Se alguém tiver uma dificuldade com o QR Code, acho que não, acho que todo mundo conseguiu.
Bom, eu queria agradecer então a presença de todos.
Well, thank you for being here. We'd like to invite you to the networking moment, since we have a little bit of time before traffic gets too bad here in São Paulo. If you'd like to stick around and enjoy it, we're going to have a cocktail just outside so that we can try some Brazilian beer for those of you who are from abroad. But let's enjoy this networking moment. I'm sure it would be important for us to get to know each other better. And we'll be back at 8:30. We'll be back at 8:30. Thank you.